A delightful morning to everyone present over here. Honorable Director of Roland Institute of Pharmaceutical Sciences, Dr. J. Surya Rao sir, respected Chairperson Srimati J. Jayalakshmi Madam, Principal as convener of today's program, Dr. VVV Ravi Kumar sir, Vice Principal as co-convener, Dr. C.H. Niranjan Patro sir, Coordinator of this program, Dr. Bisu Mohan Sao sir, the entire organizing committee, today's chief guest of this program, Professor Dr. M.D. Karvekar sir, great eminent speakers, delegates from all over the country, my dear colleagues, and all the great minds present here. On this beautiful morning, I, Ms. Deepika Rani Sahu, is pleased to welcome each and every one of you to a brainstorming online faculty development program on artificial intelligence in drug development, recent trends and future perspectives, sponsored by AICTE Training and Learning Academy, New Delhi, and organized by Roland Institute of Pharmaceutical Sciences, Odisha. It's a wonderful day and I feel privileged to extend my warm welcome to all presented here. ATAL is an initiative by AICTE which aims to set up an academy which will plan and help in imparting quality technical education throughout the country. Its main objective is to empowering faculty members for achieve goals in higher education, and this program is designed to fulfill the need to train the young generation in skill sector and having faculties to be trained in their respective disciplines. Before starting the inauguration, let me focus on few highlighting points about this FDP. The objective of the proposed FDP is to explore the various approaches of artificial intelligence utilized in drug development process. This approach augments the drug development process by reducing the failure rates of clinical trials and decreases the expenditures with increase in efficiency of drug research. The FDP will focus on current trends and applications of artificial intelligence in the area of drug development and will be helpful both academicians, researchers who are involved in designing novel drug candidates for the treatment of various disease conditions. This program will definitely meet the need of pharmaceutical industry by imparting knowledge of innovative techniques of AI related to drug development among faculty members, young scientists, and researchers, etc. Now, to mark this event, a traditional beginning, let us begin a virtual presentation of lamp lightning followed by Sri Ganesh Vandana.
शुभं कुर्वं कल्याणं आरोग्यं धनसम्पदः शुभं कुरुत्वं कल्याणं आरोग्यं धनसम्पदः शत्रुबुद्धिविनाशाय दीपज्योतिर्नमोऽस्तुते दीपज्योतिर्नमोऽस्तुते शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः
वक्रतुण्ड महाकाय सूर्यकोटि समप्रभा निर्विघ्नं कुरु मे देव सर्वकार्येषु सर्वदा सर्वकार्येषु सर्वदा सर्वकार्येषु सर्वदा
With the blessings of Lord Sri Ganesha, let's start the inaugural program. Now it's time to invite our honorable patrons Dr. J. Surya Rao sir, Director, and Srimati J. Jayalakshmi Madam, Chairperson, RIPS, on board for the welcome address. Over to you, Madam.
Namaste. Good morning to all of you. Respected chief guest of this function, Professor and Dr. M.D. Karvekar, Professor and Director, Rupanidhi College of Pharmacy, Bangalore. Respected Principal Sir, Vice Principal Sir, other faculty members, supporting staff of Roland Institute of Pharmaceutical Sciences, and also participants and delegates from across the globe. Once again, good morning to all of you. It's really a great opportunity for us to hold this program or to organize this program today for the development of our faculties, that is FDP. Indeed, it is a red letter day for Roland Institute of Pharmaceutical Sciences. In short, we call the institution as RIPS. It's really wonderful to see a very great personality as a chief guest for this program today. Sir, I am extremely thankful and regardful to you for accepting our invitation to be our chief guest for this very, very unique program, that is ATAL, AICTE's Teaching and Learning Academy. And it is being sponsored by them, and we are really thankful to AICTE for this thing. And I'm extremely thankful to the organizing committee being headed by our principal, Dr. BBB Ravi Kumar, and also the coordinator for this program, Dr. Biswamohan Sahu, our vice principal, and other staff members who united together to came forward to organize this program. During about this institute, I am the chairperson. My name is J. Jayalakshmi, and we are managing this institute since last 35 years. That is, so to say, 3.5 decades. During this entire time, we were enjoying, no doubt, by imparting the quality education. That is, our aim and objective of this institute is that I can say the sole aim and objective is to impart quality education. And obviously, quality is our passion. Once quality education means today's classroom teaching will not be enough. So there should be some added things to be taught to our people so that they will be abreast with the latest technologies, latest trends, etc. So during this pandemic situation, we are very unfortunate. We could not able to see each other. We could not able to share or exchange each other our thoughts, feelings, etc. But the only platform left for us, that is this virtual platform, so that we can see each other, we can meet each other, we can share each other, we can exchange our thoughts. So thereby, I hope that we have a set of eminent and speakers across the globe. Thereby, they will definitely share their wisdom during this entire 5 days program. That is, the subject which is selected and given is that artificial intelligence. It is a buzzing subject, and we are fortunate enough to organize this kind of ATAL program, keeping the subject burning and lively subject, that is artificial intelligence, its recent trends and future perspective. And I hope that this program will be very, very, very useful to all the faculty members who have registered for this program. And RIPS is always keen to give such kind of programs, not only yesterday and today and tomorrow also. We ensure and assure you that this kind of programs will be given by RIPS through some other means. And I'm thankful to our principal sir, who is heading this institute, Dr. Ravi Kumar sir, who is very much taking interest and showing his interest, putting all his efforts, energy, enthusiasm to organize every event very, very successfully. And I hope that under your leadership and under the coordinator's leadership, that is Dr. Biswamon sir's efforts, this program will be very, very successful, no doubt at all. And I take this opportunity to wish the the program, the inaugural session, a grand success, followed by all the technical sessions. So thank you, and thank you.
Thank you so much, madam, for your overwhelming words. Now I take this opportunity to invite our respected principal, Professor Dr. VVV Ravi Kumar sir, convener of this FDP, to address the gathering. Over to you, sir.
Thank you, madam. Chief guest for this inaugural ceremony, respected Dr. M.D. Karvekar sir, Chairperson, Roland Institute of Pharmaceutical Sciences, respected J. Jayalakshmi madam, Director, Roland Institute of Pharmaceutical Sciences, respected Dr. J. Surya Rao sir, Co-convener Dr. C.H. Niranjan Patra, coordinator of this program Dr. Bisummohan Sahu, organizing secretaries Dr. Bimlan Chaudhary, Dr. Shruti, staff and students of RIPS, eminent resource persons for this FDP, and most importantly, delegates and participants of this faculty development program. A warm and a very good morning to all of you. At the outset, I welcome all of you to this inaugural ceremony of 5 days online faculty development program entitled Artificial Intelligence in Drug Development, Recent Trends and Future Prospective. This is sponsored by AICTE's Training and Learning Academy, ATAL. I'm glad that we have received a huge response from the participants all across India. So with technology and learning experiences evolving each year, it is unreasonable to expect the faculty to keep abreast of all the latest trends and teaching strategies without the proper preparation. So in order to empower teachers and faculty members to provide the best education for students, we need to invest in the best training for faculty members. I take this opportunity to thank and congratulate All India Council for Technical Education for recognizing the importance of faculty training programs and funding these FDPs generously. The topic chosen for this faculty development program is very much relevant to the current scenario. As all of you know that artificial intelligence and machine learning are changing our world and empowering the fourth industrial revolution. So the area of drug discovery and development is also of no exception. Artificial intelligence plays a vital role in drug discovery, from target selection to clinical trials with minimum labor and maximum efficiency. AI has been an efficient contributor to drug development during the COVID-19 pandemic also. It was used to identify and replicate protein structure. So it also assisted in accelerating drug discovery through validation and virtual screening. I congratulate and thank all the participants for showing interest in this FDP, and I promise all of you that your knowledge about artificial intelligence in medical field will definitely be enriched by the end of this FDP. My sincere thanks to all the resource persons who readily agreed to share their knowledge about AI and machine learning with all of us. I'm very glad to acknowledge the efforts made by the staff of RIPS, the core members, Dr. Bimlendu Chaudhary, Dr. Jeshruti, and particularly Vice Principal and Co-Convener, Dr. Niranjan Patro, towards this training program. My heartfelt congratulations to the coordinator of this program, Dr. Bisummohan Sahu, for getting approval and also getting funds from AICTE through ATAL scheme. I must agree that ever since he rejoined Roland Institute of Pharmaceutical Sciences, the R&D and faculty training activities at RIPS have been accelerated by many folds. Also, my sincere and heartfelt thanks to the management of Roland Institute of Pharmaceutical Sciences, Madam J. Jayalakshmi and Dr. J. Surya Rao sir, who were always in the forefront to impart quality education, and also for their active involvement and support for all the academic activities that are taking place at Roland Institute of Pharmaceutical Sciences. Lastly, I'm very much thankful to our today's chief guest, Dr. M.D. Karvekar sir, a stalwart in the field of pharmacy, particularly in the area of pharma education. He has served for the profession of pharmacy tremendously in various capacities for the last many decades. No doubt his presence itself inspires people. I'm very grateful to Karvekar sir for accepting our invitation and gracing this inaugural ceremony with his presence to motivate all of us. With this, I thank all of you and wish this program will be conducted successfully and meaningfully. Thank you very much.
Thank you so much, sir, for your all-time support and constant monitoring. I would now invite our respected Vice Principal, Professor Dr. C.H. Niranjan Patro, sir, co-convener, for sharing his views about this FDP. Over to you, sir.
Thank you, madam. Esteemed Chief Guest, Professor Karvekar, Director, Rupanidhi College of Pharmacy, Bangalore, respected members of the management, Principal, Organizing Committee, and my dear participants, very good morning to all. At the outset, I would like to congratulate the management, principal, staff, and students of Roland Institute of Pharmaceutical Sciences for adding another feather to the cape of Roland in the field of pharmacy education by getting grant from AICTE for ATAL program. So my special congratulations to Dr. Biswamohan Sahu for applying and getting this grant from AICTE. I pray Ma Saraswati for blessing this program to be a fruitful training program for the faculty members. Thank you very much for giving me this opportunity to share my best wishes. Thank you.
Thank you so much, sir, for your inspiring words. Now I would request a program coordinator of this event, Dr. Biswamohan Sahu, sir, to tell some few words.
Thank you, ma'am. So, a warm welcome and good morning to everyone who are present over here. Honorable Director Dr. J. Surya Rao sir, respected Chairperson Ma'am Srimati J. Jayalakshmi, and our beloved principals are Dr. P.V. Ravi Kumar, Vice Principal Dr. C.H. Niranjan Patro sir, organizing committee members, respected delegates, chief guest of today's inaugural session, Dr. M.D. Karvekar sir, eminent speakers, and also my dear colleagues, very good morning to all of you. We are very much privileged to organize this online faculty development program entitled Artificial Intelligence in Drug Development, its recent trends and future perspectives. So this program is sponsored by AICTE Training and Learning Academy, New Delhi. So we are very much thankful to ATAL for sponsoring this training program for the benefit of faculty members, research scholars, PG students of AICTE approved institutions, personnel from government and industrial professions pertaining to the relevant disciplines. So we are very much thankful to principals of various institutions throughout the India for allowing their faculty members and students for attending this program. We have received huge applications from the participants from different states throughout India. Thank you all participants for showing your interest for this training program. We are grateful to our chief guest of inaugural session of this program, Professor Dr. M.D. Karvekar sir, for accepting our invitation and sharing his valuable time with us. We are grateful to all eminent speakers for readily accepting our invitation for sharing their valuable knowledge for the benefit of participants. I am very much thankful to the organizers for giving me this opportunity to share my views. Thank you all.
Thank you so much, sir. Roland, since the year 1986, has been continuously adding success stories to its name from being one of the best pharmacy college in Odisha. It is accredited by National Board of Accreditation and is approved as Institutions Innovation Council under Ministry of Human Resource Development AICT, Government of India. Now, without further ado, I will turn the time over to chief guest of today's event, Dr. M.D. Karvekar sir, Professor and Director of Krupanidhi College of Pharmacy, Bangalore, to share his views about this FDP. Take the opportunity to introduce her to all of us. For that, I request to Mr. Rajaram Das sir, Assistant Professor of RIPS, for the introduction. Over to you, sir.
Thank you, Ms. Deepika. Good morning, respected dignitaries and most valued participants of this AICTE Training and Learning Academy sponsored online faculty development program. It is a matter of great privilege and I feel honored to get an opportunity to introduce today's chief guest, Professor Dr. M.D. Karvekar sir, to all of us. Dr. M.D. Karvekar sir has completed his B.Pharm degree from Government College of Pharmacy, Bangalore in the year 1971. Sir has completed his master's degree from LM College of Pharmacy, Ahmedabad in the year 1979. Sir got recognized with PhD from Government College of Pharmacy, Bangalore University in the year 1990. Dr. Karvekar is having a teaching experience of around 5 decades, and this journey all started as a lecturer in Government College of Pharmacy, Bangalore from 1972 to 1980. Later on, Sir served as assistant professor in Government College of Pharmacy, Bangalore from 1980 to 1992. Dr. Karvekar got promoted as professor in the Pharmaceutical Chemistry Department at Government College of Pharmacy, Bangalore from 1992 to 2004. Later on, Sir worked as Director Academics, Krupanidhi College of Pharmacy, Bangalore from 2004 to October 2019. Sir also served as Executive Committee Member, Pharmacy Council of India, New Delhi, nominated by University Grant Commission, New Delhi from 2004 to October 2019. Dr. Karvekar is nominated by the Vice Chancellor of Rajiv Gandhi University of Health Sciences, Bangalore as Dean, Faculty of Pharmacy, Chairman, PhD Registration Committee, Rajiv Gandhi University of Health Sciences, Bangalore. Chairman, Board of Studies, Post Graduation in Pharmacy, Rajiv Gandhi University of Health Sciences. Chairman, Affiliation Committee, Rajiv Gandhi University of Health Sciences. Chairman, B.Pharm and M.Pharm Syllabus Committee 2003 at RGUHS, Bangalore. Dr. Karvekar is also the executive member of Academic Council, RGUHS, Bangalore, Karnataka. He is a member of Board of Studies, Gulbarga University, Karnataka. Member of Board of Studies, University of Goa. Member of PhD Registration Committee, Mata Amrutamai University, Cochin. Member of Board of Studies, KLE University, Belgaum, Karnataka. Member of Academic Council, KLE University, Belgaum. Member of Selection Committee, JSS University, Mysuru, Karnataka. Dr. Karvekar sir also served as Vice President, Indian Pharmaceutical Association, Karnataka State Branch, and Treasurer of Indian Pharmaceutical Congress Association. Dr. Karvekar is an invited speaker by American Association Colleges of Pharmacy to speak on pharmacy education in India held at San Antonio, Texas. Dr. Karvekar was deputed by Government of India to study pharmacy practice departments in hospital and colleges in Australia and Singapore in the year 2011. Sir has attended and chaired various national and international scientific conferences. Sir is also the member of Drugs Technical Advisory Board, Government of India, member of Board of Governors, Naipur, Mohali and Kolkata. Sir is the member International Services Program of Accreditation Council for Pharmacy Education, USA. Sir also served as at Government of Karnataka in the Department of Drug Control as a member secretary. Sir also is the chairman of B-Farm Regulation and Syllabus Committee 2014 Pharmacy Council of India to New Delhi. Dr. Karvikar has been awarded with Lifetime Achievement Award honored by All India Pharmacy Teachers Association Karnataka State Branch in the year 2003. Sir is awarded with Fellowship Award Association from Association of Biotechnology and Pharmacy in the year 2005. Shiksha Bhushan Award honored by Association of Padma Shri Awardees of Teachers of Goa in the year 2007. Fellowship Award awarded by Indian Pharmaceutical Association in the year 2016 and Pharma Vibhushan Award by Hyderabad Karnataka Academic Council in the year 2017. With this brief introduction, once again I thank Dr. Karvikar sir for sparing his valuable time, and now I invite Dr. Karvikar sir on board to address today's gathering. Welcome to you, sir.
Thank you. Thank you, Mr. Rajaram, for your nice introduction. And good morning to all our friends. Today is a very, very important day for me to be with you in this morning for this inaugural function. Respected Jayalakshmi Madam, for your nice introduction about the college and all. Dr. J. Surya Rao, a director of this reputed institution, and Dr. B.V.V. Ravi Kumar, a principal of this college, Biswa Mohan Sahu, coordinator of this program, and other teachers of faculty of this college, the faculty of various colleges who are attending this online program, the students of this college, as well as the students of other various colleges, invitees, and the resource person who are really going to speak a very, very important session, artificial intelligence, which is very, very useful in all the fields, you can say, very, very much useful. And I have seen there are very, very famous and eminent speakers within India and also outside India. I'm very much impressed by seeing the list of these speakers. And I congratulate the principal for having chosen this topic for this ATAL Academy sponsored faculty development program. Ladies and gentlemen, today the artificial intelligence, which is used by all of us today, the mobile what we use, there is artificial intelligence which is used in the mobile, or the digital instruments which are used today for today's program or online classes, what we take. Anything you take today, or even to diagnose a disease and to prepare drugs, for all these purposes, this artificial intelligence is very much useful. The drug discovery 4.0, which is a very latest drug discovery, and this 4.0 is very, very important. And this 4.0 also include this artificial intelligence. And in this 4.0, the manufacture of the drugs is so perfect that no human being is involved in that. You put a raw material and all robots, they work, and ultimately what you get is a finished product. To that level, now the pharmaceutical industry has reached to this stage. And that is why I was telling the principal of this college who had chosen this artificial intelligence topic for this faculty development program is very, very appropriate. Friends, as you know, this because of this artificial intelligence, today we have reached in such a position in the manufacture of the drugs that we stand number 3 in the world in the manufacture of the drugs by volume. And if you take a value wise, we stand on 14th in the position in the world. And the drugs which are manufactured here using this artificial intelligence, today we are manufacturing the drugs worth of about 2.7 lakhs crores of rupees of worth of drug. And out of this 2.7 lakhs crores of drugs which are manufactured in India, 1.4 lakhs crores worth of drugs we use domestically. We use for the entire population of India. And the remaining 1.3 lakhs crores worth of drugs we export to the other countries. We export these drugs to around 200 or more than 200 countries in the world. Once upon a time, we were importing the drugs, but today the situation has come because of the effort of the earlier people and because of the use of this artificial intelligence. We are exporting the drugs to around 200 countries, worth of about 1.3 lakhs crores. Friends, the situation to come to this stage, I can proudly say that the pharmacy educations have also contributed a lot into this. A college like Roland College of Pharmaceutical Sciences, which is well established in 1986, and I have seen in the Google the building, the infrastructure of this college, such a beautiful infrastructure we're having. I have seen the faculty, they have a very well reputed faculty in this institution. faculty is the one which really brings the name to the college, and the infrastructure and the facilities. All these things, this Ronald College of Pharmacy, which I have seen, they have really all the facilities over here. And the management is very keen to give the facilities to the students. So we have to appreciate the management also. I have also seen that the staff members who are involved, they show a lot of interest. For example, now this program is going on. I could see that many staff members are involved in this and they show the interest, and because of that, the program goes well. I also remember your earlier principal who has contributed a lot to this college. Of course, because of his retirement now, he is not there. Otherwise, really he has contributed a lot to this college. I'm really very happy about this. Now, coming to this drug manufacturing, which I was mentioning, that the India is such a country. During 1995, our consumption of the drug was very, very less. Today, it has grown up. So also, we were not exporting the drugs to that extent. Only around 15,000 crores worth of drugs we were exporting even during 1995. But now if you see, during 2019, we are exporting 1.3 lakhs crores of rupees of worth of drugs we export. So that shows how much, our country has advanced in the manufacture of the drugs. We have to also be very proud that the vaccines which are exported in the world, we are number one in the export of the vaccines. 60% of the world's requirement, the export is from India. And we are proud that the companies, Serum Institute and Bharat Biotech in Hyderabad, they are the main people who are involved in exporting these vaccines from India. The other country, they may develop a vaccine, but ultimately the manufacture of the vaccines, they will have to request the government of India for the manufacture of these vaccines in India. You have seen the Covishield is manufactured in India, and now Covishield is a good vaccine which is used not only in India, in abroad also. India is a country where the quality drugs are manufactured, and because of the quality and the good facilities to manufacture the drugs, the eyes of the other countries on India. The country like USA requested our Honorable Prime Minister to send the hydroxychloroquine during the shortage of these drugs in the USA. And our beloved Prime Minister, he readily agreed and he saw to it that hydroxychloroquine is sent immediately to USA to safeguard the health of the US people. Such is the country, and everybody's eyes are on India now. Because of this pandemic problems, people who used to get drugs from China, now it has become difficult to get. Rather, countries are not interested to import the drugs from China, but now their eyes are on India. And this pandemic situation is really going to bring a good fortune to India to introduce more and more number of pharmaceutical industries in India. And because of this, I am sure that our pharmacy students will have a good chance to join the industries in India. Friends, there are about 10 very reputed pharmaceuticals in the world, and their revenues are also very high. Company like Johnson and Johnson, the revenue per annum is $56.1 billion, which is a very high turnover. The company like Pfizer, the revenue is around $51.75 billion. Roche, the turnover is $49.23 billion. Novartis is $47.45 billion. Merck and Co. is $46.8 billion. GlaxoSmithKline is around $44.27 billion. Sanofi is around 40.46, and so on. So this much of revenue to these pharmaceutical industries. So also the Sun Pharma in India, the turnover is also quite high. And there are Reddy's Laboratories, which is also equally very high. Cadila, which is also very high. And because of this pharmaceutical export, which I was telling you 1.3 lakhs crores worth of drugs, the government of India is also thinking to have a separate ministry for these pharmaceuticals. Now the petrochemicals and pharmaceuticals, it is both are merged, and there is one ministry now. They want to bifurcate it and make petroleum a separate ministry and pharmaceuticals a separate ministry. And if this happens in a short time, I think the pharmaceuticals will have still a brighter future. If you take the Indian companies, the top Indian companies are Sun Pharma, Reddy's Lab, Cipla, Divi's Lab, Aurobindo Pharma, Glenmark Pharmaceuticals, Biocon, Lupin, and so on, Zydus, and so on. These are all very important, industries in India, which are contributing a lot. If you take the Sun Pharma, for example, it established in 1983 in Gujarat. And today, in 2019, if you see, the revenues are 30,091.4 crores. The revenue, that is around 4.2 billion US dollars. And if you see the employment in this Sun Pharma, There are about 32,000 people are employed. So many people, see this number. It's quite a big company. If you see the Reddy's lab, the Reddy's lab is established in the year 1984, and the revenue today in 2019, if you see, it is 17,460 crores of rupees, that is around 2.4 billion US dollars. And the number of employed people in this company are around 21,966. So next to Sun Pharma is Reddy's lab. The Devi's lab, which is started very, you can say recently, we started in 1990, and the chairman of this was a student of Manipal College of Pharmacy, Manipal, Karnataka, during our time. And he has given employment right from his student days. He was telling that he doesn't want to work, but he wants to employ. And that, he has really made it. Around 15,000 people are working in this company. Like that, there are many companies which have come up and they have given a good employment and good business. And because of this, the government of India, just like software companies, now they want to have a separate ministry for these pharmaceuticals. To grow these pharmaceutical companies in India, we have to thank the earlier people who have really helped to bring our Indian pharmaceutical companies to this level. The first and the foremost, I would like to thank and remember one chemist in Kolkata, J.C. Ghosh. In 1918, he is the first person to fight that there should be a regulatory body for this drug business. Because during that time, there is no any regulation. You can sell anything and there is no any check also. But he thought drug is an important, and that is why we should have a regulatory on this so that the quality drugs should be available to the public. And he is the first person to start the fight against to have the regulatory people for control of the quality of the drugs, that is Mr. J.C. Ghosh. Because of his fight, the government thought that we can have a committee, and R.N. Chopra committee came in 1930, and the Chopra committee recommended that there should be a qualified person to manufacture the drugs or to sell the drugs. And because of his recommendation, the Drugs and Cosmetic Act came in 1940, and then Pharmacy Act came in 1948 to regulate the pharmacy education. The first regulation of the pharmacy came in 1953, ER 1953 for diploma. Then the next regulation came in 1972, and after that, Education Regulation 1981, and then Regulation 1991. After this Education Regulation 1991, diploma, what is going on even today? The Pharmacy Council of India is pleased to have a new regulation, Education Regulation 2020, and I am sure that already the council has circulated the comments on this syllabus, and your college also must have received this regulation. And I am sure that if everything goes okay from this academic year 21, the Pharmacy Council of India wants to introduce this new regulation to 2020 in 2021. And that will be an entirely different regulation. The syllabus is entirely different what so far we were studying. This is a practical oriented syllabus, very beautiful, and many educationists, they joined together. They have contributed a lot to make this new regulation, and I am sure that the students of diploma in pharmacy, they will really enjoy this new regulation. Friends, there is a lot today which we want to see about this artificial intelligence which has contributed, and because of the contribution of this artificial intelligence, we could reach to this level. The Pharmacy Council of India is working very hard to give the best out of it. The dynamic president of the Pharmacy Council of India, Dr. B. Suresh, who is working so hard to uplift our education system to a very, very high standard. He brought so many regulations. We had earlier only regulation for diploma, and there was no any regulation to control the degree and post-graduation. First and the foremost thing is that we brought the new regulation, 2014 B-Farm regulation, 2014 M-Farm regulation, and then they have brought the regulation for teachers, the regulation for practice. In 2008, Form D regulation, which is a baby of our president. When he visited USA, he saw it that the Form D course which is running in the US, and they have stopped this B Farm for the entry. He thought that our B Farm graduates which are coming out in India, and if they are not accepting, US is not accepting these B Farm graduates for employment purpose in USA, then our students, they lose the chance in going abroad for the job. First thing what he did, that the course which is run in the USA, the same course, why can't India starts here? So he called the experts from US, he called the educationists in India. We had a meeting, a marathon meeting, and then we decided that in India we should start the Form D course. And since it is a course which is mostly the hospitals are involved, so we made the regulation that a college which is having a hospital of its own, or maybe their sister concern hospital, or maybe a private hospital, where they can take a MOU from that hospital to run this course, and a hospital which has got 300 bedded. Now, such colleges, we gave the permission, and today the Form D colleges in India, they cross about 200 colleges in India. This Roland College of Pharmacy, which I have seen, they have D Farm, they have B Farm, M Farm, and maybe PhD they must be having. But then, if they can start this Form D course, I'm sure that the students who are nearby, they will get a benefit from this course and they will have a better future in the practice. I'm sure that the madam will take an interest and she will definitely, think on this to start this Farm Day course. Then, this Farm Day course background, we wanted to start. During that time, in the US, the American Association of the Colleges of Pharmacy, ACPA, they invited me to give a talk on pharmacy education in India. I attended that meeting in San Antonio, Texas, and I was talking to them about the pharmacy institutions in India, and we had introduced the PharmD course in India. When they heard that the India also has started the PharmD course, they really shocked. They were thinking the software companies, they have gone ahead so much in their field, and now the brain from India, if they start this PharmD course, they overtake the US in this practice. This is what they were discussing. But then we said that we have only restriction for the intake, that is only 30 seats. Whereas in the US, PharmD intake is 200 and more also. They comparing it is only 30. But when we say that degree we are having around 2,000 colleges, they were also really shocked. This PharmD course also may increase the number, and PharmD people in India, it will be more in number. They were telling that they have number of jobs for PharmD over there. They're not able to, cope up with the vacancies that were available. And I told them our students are there ready to work, come and work in our country. And that is how, now our students, they have started going USA and they started going to Australia for the job. Friend, there is a lot to be talked about the pharmacy course and the working of the Pharmacy Council of India. The Pharmacy Council of India is also now thinking that our teachers who are teaching this PharmD course as well as the B Pharm and M Pharm courses, they lack the industrial experience. And the Pharmacy Council of India wants to depute those who are interested to go to industry for 1 year. Our President, Pharmacy Council of India, has already contacted many of the industries and he has discussed about this issue, and the industry people are very much interested to train our teachers in the industry so that they can gain the knowledge from the industry. They can also see how this artificial intelligence works in the industry. They can come back from the industry and they can guide these youngsters, because ultimately, whatever we produce, these students, they will have to join the industry for a good job. And industries are telling that they are not getting a person whom they want to employ. But when our teachers are trained, and if these in turn the teachers, if they train the students, we are giving the students to the industry what they wanted. And this academia and industry interaction will produce the good students who are employable in the industry. And that day is now going to come very near. And I am sure that the institutions will cooperate with the Pharmacy Council of India and make our president's dream a real one. With these few words, I thank the management of this a very reputed college, Roland College of Pharmaceutical Sciences. And also I thank Ravi Kumar, who called me for the first time, and then he requested me that, sir, please, you'll have to come. And I really agreed. I thought that we should, tell what is happening today to all our students and our faculty, so that they can take up certain positive points and they will pass on to the students. With this few words, I thank one and all. Thank you very much. You have to unmute, you have to unmute. Yeah, good.
Thank you so much, sir, for sparing your valuable time with us and grace this occasion with your presence. Thank you so much, sir.
Thank you.
Now I request to Dr. Bimlendu Chaudhary, sir, organizing secretary of this event, for the concluding remarks and vote of thanks. Over to you, sir.
Good morning, one and all. Myself, Dr. Bimlendu Chaudhary, Professor, Roland Institute of Pharmaceutical Sciences. Very much thankful to the organizing committee for giving me the opportunity to propose the vote of thanks. First of all, I would like to give my hearty vote of thanks to our chief guest, Professor Dr. M.D. Karvekar sir, Professor and Director, Krupanidhi College of Pharmacy, Bangalore, and Executive Committee Member, Pharmacy Council of India, New Delhi, for sparing time from his busy schedule to grace today's inaugural session of ATAL Online FDP program. I also thankful to the guest, to the members, or to the that means distinguished guests of today's function for giving their valuable time to attend this particular inaugural session. We are very much thankful to our beloved director, Dr. J. Surya Rao sir, and chairperson, Madam J. Jayalakshmi, for their presence and constant support and motivation for organizing such kind of program. I would like to express our deepest gratitude to our principal, Professor B.V.V. Ravi Kumar sir, Vice Principal, Professor C.H. Niranjan Patra sir, for sparing their valuable time to attend the meeting and constantly encouraging for organizing such type of events. I would like to thank the program coordinator, Professor Vishwamohan Sau, and co-organizing secretary, Dr. J. Shruti, for their precious effort for organization of the program. I also extend thanks to ATAL Academy AICTE for selecting our institution to organize this online FDP program. At last but not the least, I thank from the bottom of my heart to all the participants for registering for this online FDP program. I hope that all the participants will be immensely benefited by the oncoming lectures by our eminent speakers. With the warm words and kind message from our chief guest and our dignitaries, we officially conclude the inaugural ceremony of this ATAL online FDP program. I wish this FDP a resounding success. Thank you all.
Thank you so much, sir. Now I request to our beloved chairperson, Srimati J. Jayalakshmi Madam, to present a memento to Dr. N.J. Karvekar sir as a token of love and appreciation on behalf of the entire Roland family.
Sir,
Thank
thank
you.
you very much for
Thank you
your
so much.
valuable presence today,
Thank you.
and we
Thank
are
you.
deeply immense and indebted to you, sir.
Thank you, madam. Thank you.
On behalf of the management, principal, vice principal, staff and students, and as a token of our respect and memory, we present you the certificate and memento. Please accept, sir.
Thank you, madam. Thank you so much. Yeah. Thank you.
Thank you very much, sir.
Thank you. Very nice of you. Very nice of you.
Very nice, sir.
Thank you, Ravi Kumar.
Thank you, sir. Thank you. Thank you very much.
Bye. Please carry on.
Thank you, sir.
Once again, a very good morning to all the dignitaries and delegates present over here. I, Ms. Deepika Rani Sahu, Assistant Professor, RIPS, on behalf of RIPS, again welcome you all to the first session of online FDP program on artificial intelligence in drug development, recent trends and future perspective, sponsored by AICTE Training and Learning Academy, New Delhi, and organized by Roland Institute of Pharmaceutical Sciences, Odisha. Here we have a great eminent speaker, Dr. S. Sivasatya Madam, with us. Today, Madam is going to deliver a lecture on application of artificial intelligence for healthcare and drug design. Before inviting her to board, I take this opportunity to introduce Madam among all of us. Madam is currently heading the Department of Computer Science in Pondicherry University. She has 21 years of experience in teaching and research. She has, to her credit, 36 international conference publications and 70 research papers in reputed international journals. Her research interest includes evolutionary and bio-inspired computing, machine learning, spatial data mining, bioinformatics, and vehicular ad-hoc networks. She is the recipient of the prestigious Nari Shakti Puraskar in 2017 from the Honorable President of India for developing the Women's Safety App Mitra. Also, she has obtained the Chairman's Distinction Award in the South Asia's Brilliant Award in 2017 for mobile innovation. She is involved in several committees in close association with the government. The RoboCo in the Puducherry beach is also done by her students under her guidance. She is also part of several projects for the Puducherry Police and JIPMAR. With this brief introduction, I welcome you, Madam, and request to continue the session.
Thank you for the nice introduction. Very good morning to all and all present here. I'm really honored to be here, and I'm happy to share my expertise, whatever, to the esteemed audience. But I'm very sad that I do not know how much time they'll give me to share my complete lecture. Sir, till what time I can proceed?
Yes.
I think I'll have
Yes,
to
yes,
touch on.
ma'am. No, ma'am, you can take your own time.
no, there's another speaker. I think like at 12 o'clock. So it's already 12. So till what time I
Yes,
can
yes,
go?
no
Five
problem.
p.m.
No,
Now
Okay,
we'll
sir.
no
request the
problem.
speaker.
Okay, sir. Okay. So thank you for the opportunity given. So I think I'll share my without wasting much time. I'll share my presentation. Can you see my screen?
No, ma'am.
Oh, okay, one second.
You go to share screen.
Yeah. Because this is
Yes, yes, just came, ma'am. Just came.
just go to the presentation.
Make it full screen, ma'am. Yes,
Is it
make
okay,
it
sir?
hide. Yes, yes, just make
Yeah.
it hide, ma'am. Hide.
Yes.
Yes, yes. Continue, ma'am. Thank you.
Okay. So today, my topic of discussion would be application of AI for healthcare and drug design. Um, so the agenda would be like this. I'll be just giving a brief introduction about what is artificial intelligence, general perception that people have regarding AI, key areas where it could be applied, and application. So that involves your drug design and what kind of AI techniques used and so on. So, coming to the topic, what is intelligence basically? So, everybody has got intelligence. We know, like, we are all intelligent, and that is what differentiates us from the animals. So, we have the sixth sense, right? So, the ability to comprehend, to understand, and profit from experience. So, experience makes the man, right? So the more and more you are experienced, you get to have more knowledge. That is what people say. So whatever you learn in your classroom, that might not be very much sufficient, but once you get or get yourself exposed to the open world, then you learn a lot of things, and with age this goes on and on because it gets accumulated. Maybe it is like direct learning, or it might be an unconscious assimilation of concepts. Whatever you look around, whatever you see, whatever you understand, whatever you speak, so everything goes into your head without your knowledge actually. So it goes on accumulating, and that becomes your experience actually. So it's a general mental capability that involves what are the things that it involves. Let us see the ability to reason. So you should be able to reason out why we are doing things, why we are doing this, and why he's doing that. So ability to reason, plan. So plan out something for the future and solve problems. Think think abstractly. So everything we cannot go in depth and think. Say for example, if you see say for example pharmaceuticals, so that is actually abstract for me. I do not know much deeply into that, but I should be able to think abstractly like what it is actually and how I can apply my AI technique to that. Then apply common sense. So certain things are not explicitly mentioned to you. Everything will not be okay. I mean, if you say like attend the seminar, nobody will come and tell me like, okay, you have to do this, you have to open your laptop, you have to click on the link, all those things, like it's just applying common sense. Recall past experience. So I would have conducted various meetings. So maybe this StreamYard is new for me, but still I have conducted various meetings through Zoom, Google Meet, WebEx, and so many things. So maybe I just had to recall those past experience and then learn from it. So perceive, interpret, and respond to the environment. So this is what is basically call it as intelligence. Clear? And so, what is artificial intelligence? So, artificial intelligence is making the machines learn. You are trying to incorporate or imbibe or imbue, like you are going to give some intelligence to the machines. That is what is called as artificial intelligence. So, John McCarthy, like he's widely recognized as one of the godfathers of AI. So, he defined it as follows: like it is the science and engineering of making intelligent machines. So, you want to make machines intelligent. So previously we had computers, like people have been using computers for a long time, for several decades together. But are they not intelligent? So that will be your question, right? So are they not intelligent? Like whatever we say, like it's doing, no. So is it not intelligent? But according to intelligence, like for example in the previous slide I told you, not only the ability to do, like even you may have a dog or you may have a servant, like he simply says like whatever you tell it will be doing like a slave. It's not like that. But you should be having the ability to think on your own, solve problems on your own, apply the common sense and recall the past experience and learn. So all these things should be incorporated. So that is what like we need to give it to the machine. So previously the machines were like calculators. They were doing like whatever jobs you gave them to do, or you just ask them okay to calculate the employee payroll, or just like calculate my BMI given the height and weight, or like even now you have the smart watches, like wherein you say that it is an AI enabled watch, but it is not so. Like it's again like a basic machinery which calculates the body mass index based on a thing, based on your height and weight and based on your step count. It says like how many calories. So it's all like any calculators can do. So it's not, you cannot call them as intelligent. So what is intelligence, what is artificial intelligence is you have to make the machines think and do on its own by learning from the large amount of data set that you expose the machine to. So you have to expose the machine to a large amount of data set by which they start learning on their own. So that is what is called as artificial intelligence. So isn't it expert system? So you'd have heard about expert systems or decision making systems. Is it so? So what are expert systems? So you would have heard of many expert systems actually. So expert systems are nothing but systems which can give you some solutions for your answers. So or you have a lot of decision making systems, like so if I have this much of temperature and if I have been having headache for this many days, like okay you might be having fever, so kind of a decision making system or an expert system. So are they AI? Your health monitoring systems, your smart watches and other fitbits and all those things, are they AI? So are they all having intelligence? No, it's not. It's a big question. They might not have intelligence. They can do it without intelligence also, because they have been programmed to do something routinely. If you ask them something new, they cannot be able to tell, because they cannot recall their past experience and they cannot apply that to this. Okay, so they have been programmed to do something and they will do that efficiently. So that is not artificial intelligence basically. So artificial intelligence involves something more than that. So there are so many things in artificial intelligence. Artificial intelligence involves like automated reasoning. You can see this figure, automated reasoning. You have computer vision, like the system or the machine should be automatically able to look into something and identify pictures, identify images, identify the activities carried out. So all those things, it comes under computer vision. Automated reasoning, like once it has done something, a similar task in the previous time instance, it should be able to do a much better thing in the future. So that's automated reasoning. And machine learning, autonomous systems, natural language processing. So if I speak in English, the machine will not understand, but it should understand. So that is the branch of natural language processing. So even if you talk in the natural language, any language, say for example in Google, you can have Google translators, and even if you tell in English or Hindi, it will accept, isn't it? So there is like lot of natural language processing that is underneath the system. And then intelligent agents, deep neural networks, so all these things comes under AI. So AI is a very big umbrella term under which you have so many sub branches, like you have all these things coming under that: vision, computer vision, machine learning, deep learning, intelligent agents, NLP, there is natural language processing, autonomous systems, automated reasoning. So all these things comes under artificial intelligence. So artificial intelligence is not one single area. So it involves a very very broad spectrum that encompasses so many subdomains, and each is a ocean actually, if you see that. If you have any doubts, or if I'm going too fast, you can break in between and you can very well raise your questions. I'll be happy to answer you. Is it okay? So again, like this is yet another thing of division. Like artificial intelligence, you have neural networks, data mining, statistical AI, pattern recognition, swarm intelligence, evolutionary computation, expert system. So we will be seeing later about this. So there are so many classifications. That's what I am just trying to show you. So artificial intelligence is an umbrella term which covers all these things. So you have, so people talk about machine learning. So whenever AI comes, it doesn't go without machine learning. So machine learning, deep learning, and again deep learning is a subset of machine learning. So under artificial system, you have expert system, NLP, speech recognition system. You have planning or decision making systems, robotics, computer vision. So under vision, you have image recognition and machine vision, two branches. Under speech recognition, you have speech to text and text to speech. So I now whatever I speak, it will be converted into text, or when I give some text, it will be converted to speech. So there are so many applications of each of this into, for different areas. And if you come to natural language processing, you have text generation, question answering, context extraction, classification, machine translation, and so many other things. So you can see some of the AI definitions here. So it's a branch of computer science dealing with the simulation of intelligent behavior in computer. So we are just simulating. We cannot make the machines completely intelligent, isn't it? So we can only simulate our intelligence. So mimic it. So the capability of a machine to imitate intelligent human behavior. Or you can say that a computer system able to perform tasks that normally require human intelligence, such as visual perception. So what do you mean by visual perception? Looking at something, you should be able to perceive what it is. What is your perception of that image? Say, for example, if I can understand that a boy is playing ball. Okay, suppose the machine sees it. So using a camera, you capture the boy who is playing ball. Whether the machine will be able to say that the boy is playing ball, automatically it cannot say, isn't it? So it has to be programmed. So how it has to be programmed? So it has to be given lot of training. Say maybe in one post, it will say, like if I give only one image of a boy playing ball, it can say, okay, boys playing ball in the later instance. But similarly, today he'll be playing volleyball, tomorrow he might be playing basketball, or he might be playing cricket with the ball. So all these things are involving ball. But whatever be the kind of a thing, it should be able to say that the boy is playing a ball. So this kind of a visual perception, how does it get actually? So how does it get? In the sense, it has to be trained with a large amount of data set. So data set has to be very, very large. The training data set that they call it as. So only then it will be able to do the visual perception. Then the speech recognition. So whatever language you speak, it should be able to understand. So for that also you have to train the machine. And then coming to decision making, at different instances of time or different points of time, like how to decide on something, like the human beings, and translation between various languages. So that is also required. So these are some of the definitions again. AI can be a pile of if-then statements or a complex statistical modeling, mapping raw sensory data to symbolic categories. So what do you mean by if-then statements? Actually, in computer science or computer science programming languages, normally the code is written with the case of if-then statements. Like if-then means, like if this condition is there, you have to do this. Say for example, if say you want to count, or if you want to search for element in a big list, so in that case what you will do, no, you will give a condition. If the if element is equal to 100, then fine, you report that it is found. So likewise you can give if then statements in a language. So AI could be a pile of if then statements. So you can have a number of if then statements that is giving a reasoning capability to the machine. That is like, if this happens, you do this. If this happens, you do this. If this does not happen, you do this. Or you come out of the program. So likewise, it could be a pile of if-then statements or a complex statistical model. So depending upon various statistics, you can have make it a complex statistical model mapping raw sensory data, whatever sensory data you collect from the surroundings, to symbolic categories. Whether you classify that as something, or you say it's a high risk of this disease, and so on. And the if-then statements are rules that are explicitly programmed, also called rule engines or expert systems. So previously the AI were only expert systems, they call it as AI. So instead of meeting a doctor, if you had a number of expert systems previously which could actually answer to your query. Say, okay, it'll ask like if you want to check if you have fever or if you want to check if you have COVID. So an expert system will be like this. So how it will be like, okay, do you have temperature? Yes. So what is the temperature, whether it's in this range or not? Okay, if you give yes, then it will go to the next question. It will ask, okay, did you have headache? Yes. If you, then it will go to the next. Did you have throat pain? So if you say no, then it will go like different other questions it will ask, and accordingly it will decide whether you have COVID or not. So this is a simple expert system. So this will be a set of if then statements or knowledge graphs, or you can say it is a symbolic AI. So these are all termed as good old fashioned AI. So now it is not, it's all old now. So now the AI has made rapid advances in leaps and bounds. It has taken different shapes. So these are all old fashioned AI. And now people talk only about machine learning and deep learning. So what is machine learning basically? So because you're going to hear this terminology almost everywhere you turn around, because machine learning is the talk of the day, machine learning and deep learning. So machine learning, if you see, it's just a subset of AI. So that's one field of AI. So symbolic logic, rule engines, expert systems, and knowledge graphs could all be described as AI, but none of them are machine learning. You cannot say those are all machine learning, because you're not teaching the machine to learn. You're only giving some conditions and actions to be carried out in a particular context. So that is what is rule engines or expert systems and knowledge graphs. But AI, machine learning is something different. So in 1959, like Arthur Samuel, a pioneer of machine learning, defined machine learning as follows. So what do you mean by machine learning? So it's like a field of study that gives computers the ability to learn without being explicitly programmed. So you should note here, it is explicitly programmed. So you will not be intervening at each point of time and explicitly say, okay, do this, if this condition, do this, if this condition, do this. No, you're not going to tell that. So if you tell that, then it will become a expert system or a rule engine. But here it will be like a child. How does it learn? So when it is when a baby comes to this earth, does it know everything? It will not know. Slowly it will learn to recognize colors. Slowly it will learn to recognize alphabets. Slowly it will learn like, okay, fruits. So how do they teach in your kindergarten? So when a child is sent to the school, they teach him by showing all this, no, all the toys. So they'll take, okay, for colors they'll have a chart. They'll say this is green, and similarly, to understand that it is green and not the shape, it has to, they'll be showing different green objects. This is green, 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 green. So red, red, red. So likewise, they'll teach colors. Shapes, they will teach shapes slowly. By showing all the shapes, it is slowly learn. Similarly, fruits means they will show all the fruits, how to eat, and so like something it is an eatable. So like that they will show, no, just by showing so many things, they will try to put everything into its head. So that is basically what is machine learning. So the more and more you show, the more and more you this is what I call it as a training data set. Is it clear? So this is what I can call as a training data set. I train the kid with all kinds of shapes, figures, objects, eatables, so all those things. So the more and more I show, the more knowledgeable the kid will become. So similarly, to the machine, I have to give more and more data set with a label. With a label in the sense, okay, this is what this is a car. Car means all the cars I will list out and I'll give the names. So that means like if I show you a new car, it will be able to say like, okay, this is not in the list, like this is a new car, it's a new model. So kind of I'll be able to do. And similarly, I'll be showing different kinds of people activities like playing, crying, smiling, laughing, all kinds of things I'll be giving. So a field of study. So that is what machine learning. So machine learning is something like is akin to a child's learning from his small age, from his childhood. Okay, so that is why I've shown this figure. So a field of study that gives computers the ability to learn without being explicitly programmed. So machine learning programs, they adjust themselves in response to the data they are exposed to, analogous to your child's learning. So as the child grows, You did not tell him explicitly everything. Even if you show him a new fruit with certain characteristics, it might recognize, okay, this is also a fruit which I can eat. So that is what it has to be done in the missions also. So machine learning programs adjust themselves in response to the data they're exposed to, analogous to your child's learning. And experience for machines is data input versus result accuracy. So the more accurate results it gives, that is good actually. So that is like how much data you give, that much accuracy you will be able to get. So that is what you mean to say by machine learning. And the learning part of machine learning attempts to optimize the output while minimizing the error of prediction. So it keeps on predicting what is what, and that the learning part of the machine learning will be attempting to optimize it. Every time it will try to better the best. So how to reduce errors? Like that's what like neural networks do. So we talk about neural networks. So that is again a branch of this, and that is what how to reduce the errors in prediction. So that is done by the neural networks. So they keep on measuring the error and modify the parameters until they can't achieve any less error. So this is what. So neural networks, if you see, they are again a set of algorithms, and they are modeled loosely after the human brain. Okay, you can see that they are algorithms, they model mimicking the human brain. They are that are designed to recognize patterns. So patterns here recognize your numerical contained in vectors, like it could be images, sound, text, or time series mapped to vectors. Vectors means it's an array, it's a list. All numerical coordinates, they'll do some conversion process and give it to the give as vectors to the machines for understanding or learning. And neural networks help us cluster data and classify data. Say, like for example, disease. Say you have several X-ray images, for instance. So X-ray images of COVID patients and non-COVID patients. So if you have thousands and thousands of such X-rays, you can give it to the machine, and you can say like it will be able to, you label it. This is COVID. This is non-COVID. So if you have 1,000 images of COVID patients and 1,000 images of non-COVID patients, so you write some algorithms to train the machine to understand the patterns that are involved with COVID and non-COVID. So tomorrow, if you give a new X-ray, it will be able to identify whether it is a COVID or non-COVID patient. Okay, so that is what neural networks do. Neural networks helps to cluster and classify. So you can say like you can give, it can cluster a group of data according to certain thing, and group unlabeled data according to similarities among the example inputs. So you're giving an example inputs. You're giving thousands of COVID patients X-rays and thousands of non-COVID patient X-rays. So in that case, from the examples, it is able to learn the patterns, and according to the patterns, it is coming to a conclusion. Okay, if the X-ray is like this, then it is COVID. If the X-ray is like this, then it is not COVID. And that is what you call as learning. Okay, and they classify the data when they have a labeled data set to train on. So it can also extract features that are fed to other algorithms for clustering and classification. So it will try to find out like what set of features are important to designate this particular X-ray as a COVID or non-COVID. So it will also try to extract features on its own. So this is a simple neural network. It's just represented as an input layer. You give the numerical input, and that will be given to a hidden layer. So it will do some mathematical processing, and then you have a output layer. So this is like why it is called as a neural network. Is like every so you call the circles here as neurons. Say input neurons, output neurons. This is like just like your brain. The neurons in your brain, so they're all connected with so many edges, and the weights and the parameters that are given to this will be adjusted accordingly, and the output will be obtained. So if you have a number of layers like this, then you call it as a deep learning neural network. So here you can see that there are a number of hidden layers, and these hidden layers will actually serve to do more complex processing, identification of better and better features, and do more complex tasks that are involved in language translation, computer image recognition, and so on. So this is a deep learning neural network. And deep learning is a subset of machine learning again. So AI, then you have machine learning, then deep learning. So it's a better and better. Uh, uh, I mean, it is very very popular now. And the term deep learning refers to deep artificial neural networks. So it is inspired by the structure and function of the brain. And deep, why deep means it refers to the large number of layers. So this is actually a simple one that I have shown here. So you'll have like hundreds of layers also. So depending upon like how many features you have to extract and what is the application that is underneath. Clear? So deep refers to the large number of layers and improved accuracy for many important problems such as image recognition, sound recognition, recommender systems, natural language processing, for all these things. So you can see this graph here. So the performance of, say, like for example, if you are machine learning and other algorithms, they are their performance will slowly go up and finally get stuck up in a plateau. So that is like it is flattened. It will not improve further to a point. But deep learning, as and when you increase the amount of data given, so the more learning it does, and so the performance will improve like this. So that is why, like for deep learning, you need large amount of training data set to be given in order to make it function very well. Okay, so that is so this is something like, say for example, here they are given, these are 1980s era neural network, very simple. One hidden layer was there, one input layer, one output layer. So it was a very simple one. But now it is like, it is so many layers you have. Say for example, if you give one input like this, so looking at this input, the machine has to tell who it is. So to tell how it is, that is image recognition it has to do. So it will break this image into so many pixels, and maybe which region, it will take different regions of this, identify it. Like first layer will identify all the edges in this, second layer will identify the combination of edges, third layer will identify features, and fourth layer will, so it's not four layers, like it will be more, identify combinations of features, and finally it will say output is George. So this is how it does, because like you might be thinking what, it's just a simple recognition, but to recognize a person, how does the machine know how to recognize? So for that, lot of work has to be done underneath. So that is being done by various programs that are learning underneath. That's a deep learning neural network algorithms and functions will be doing that. And say for example, from your side, like you can say it's a deep neural network, it's a tumor. So input layer, then hidden layers will be processing, and the output layer, you have the tumor in the image is a oligodendroglioma. I do not know much. The tumor is, the image is an what kind of a tumor also it will say, because would have trained the machine with different kinds of tumors. So thousands of images. So here the scale is like not one or two images. If you train, then it will be like a child who is not exposed to the outside world. If you give more and more number of images, it'll start learning better. So that is the essence of deep learning. So it will say like what kind of tumor it is, and not only two classification, it's a binary classification you call this is, it will have more than that. Okay, so that's about the simple introduction to deep learning and machine learning. If you have any questions, I'm open, or I'll go to the next. Any questions? This is a small introduction to AI I have given.
And continue, madam. They will share the question on chat box.
Okay. Chat box, I can see only at the end
No,
because I have my.
okay, no need. No need. You continue. Lastly, we can discuss.
Fine.
Thank you.
So now we'll come to AI in healthcare. So how AI has influenced the healthcare industry? So because artificial intelligence has been used throughout, like in all domains, in all walks of life, we can use AI. But how does it impact the healthcare industry? How does it influence the healthcare industry? So AI simplifies the lives of patients, doctors, and hospital administrators by performing tasks that are basically done by humans, but in less time and at a fraction of the cost. So, how it simplifies the lives of patients, doctors, and hospital administrators. That is what we want to see, because it is capable of doing, say, for example, if you want to check whether a patient is COVID or not, like with the pandemic that has come, doctors will get fatigued, isn't it? They will become tired. So, how many X rays they can look at per day? So it is so difficult. So at that time, we can have an AI to inspect lakhs and lakhs of what to say x-rays and simply give a report. So you can do it in less time and at a fraction of the cost. If only thing is the accuracy, we need to program in such a way that it is accurate enough. And so AI is reinvigorating modern healthcare through machines that can predict, comprehend, learn, and act. So it should be able to predict diseases, diagnose diseases, understand what it means, learn accordingly, and act. Okay, so why healthcare is in the limelight? So why now people talk about healthcare industry for AI, using AI? So why it is in the limelight? Why it is so popular? Because the amount of data that is used in the healthcare industry has increased massively. So it's a massive increase in healthcare data, and complexity of healthcare data is high, and maturity of AI field. So we'll see one by one. So the massive increase in healthcare data. Say like the average person is likely to generate more than 1 million gigabytes of health related data in their lifetime, equivalent to 300 million books. Like it's not like everybody will not have a case sheet or a record, but everyday details, like every day how is BP is increasing. So you may not think, okay, I have only one small report. No, how do I get this much of data? But if you continuously monitor a person, either his disease or not, but if you keep on monitoring every day his lifestyle, his behavior, his blood pressure, his everything, that will create around 300 million books. So that much of information you can generate from a average person. So because with the advent of all these fitbits and other things, the amount of data has increased. So IBM Watson Health has given this statistics. And the complexity of healthcare data. Say, for example, if you're working with images, okay, fine, that is a different field. If you're working with text, that is a different field. But healthcare data involves all these things, right? It will be structured or unstructured. Maybe the doctor will be writing some prescriptions, he will be generating some lab reports. So each and everything is not a structured thing. Structured means like in the form of some tables, a well-defined table with proper attributes and so on. That is what you call as structured data in computer science. Unstructured data means like just handwriting. So even we will not be able to understand the writing of doctors, isn't it? Handwriting of doctors. So they scribble something like in a prescription. So the machine should be able to read it. Means it's a very very challenging task. So you have to train the machine with millions of doctors' handwriting. So that is unstructured. Structured and unstructured you have. Then you have text, so reports and other things. Pictures you have like uh, video, I mean your scan and your x-rays, all those things are pictures. Then you have audio recordings, then you have some video recordings of operation and other things. So those are all called video. Like they take ECG, EEG data, x-rays, scan reports. So they're all different forms of data. So you can call them as big data because different forms, it's so voluminous, forms are different, so you can call it as big data. Or you can call it as EHR in different forms. EHR is electronic health record. So electronic health record comes in different formats as well. So it becomes so difficult to uh, comprehend all this data, different kinds of data. So that is why you say the complexity of healthcare data is very high. It's so difficult to understand the data, and it is difficult to train the machines with that. So that is the second thing. And maturity of AI. So AI has matured a lot compared to the previous days. Like you have artificial intelligence, I told you, no, you have predictive analytics, text to speech, image recognition, expert systems, language processing, robotics, all these things have come up. So you have machine learning, neural networks. Under deep learning, you have CNN, that is convolutional neural network for image processing, recurrent neural network for time series analysis, LSTM, that is long short term memory. And different architectures keep evolving day by day, and that is one more reason for using AI, because in healthcare we need more accuracy and precision. We cannot compromise on the quality of the results obtained in healthcare, unlike the other fields like wherein you can compromise little bit of precision. This is like health, like life and death situation. So you cannot compromise on the, say, you cannot say like a person without any disease, you cannot say that okay, you have cancer. So that is like life threatening. And if a person is having cancer, and you cannot say you don't have anything, because that is also risky, because he will not take the treatment. So this is like the accuracy has to be 100%. So that is what everybody targets with, and that is possible only with the maturity of the AI fields. And the availability of, you have open source libraries or data sets, that is a Python libraries, PyTorch, TensorFlow, Pandas, all these are some open source libraries or data sets that are available, with which you can do lot of work in AI. So why this is like, what do you mean by this? Maybe you might not understand at this point of time, because these are the libraries that are available in a Python language. Python is an open source language which is used for machine learning and deep learning. So what it does is they have contributed a lot. So people in previous days, like people would do some programming and they'll keep it for themselves, they'll not share it. But now it's not like that. Everything is put in an open source library. In the sense, like anybody can take and anybody can use it. They can do some modifications, improvise, and they can themselves put it as an open source. So all these are available open source. They are free of cost. So that is another reason why it has picked up. Okay. And above all, it's business. So business, it's a quality healthcare. So everybody wants to have quality healthcare. So leading industries have invested huge amounts of data, huge amounts in healthcare industry. And the quality of healthcare was a top priority in developed countries previously, but now in India, the quest for quality healthcare has taken over, and people are very much, I mean, very judicious in choosing a particular hospital or particular drug and so on, because they need to know all kinds of, they put in all kinds of questions in order to before taking a particular drug or before taking going to a particular hospital. So they emphasize more on the quality attributes now, isn't it? So not like olden days where people were not very familiar about the kind of treatment that is given, but now every day, like everything comes in WhatsApp, everything comes on YouTube. So everybody knows like what they are doing. Even if you give a drug, they first think about like why it is given, whether it is harmful or whether it has got side effects. So this Covishield and Covaxin and all this drugs has been designed for vaccination purpose. But how many people think that they have taken it without questioning? So still like there is a stigma. So what will happen to me if I take? So lot of things are there. So people are very quality conscious, and they keep questioning at all levels before adhering to a particular thing. So that is also another thing. So quality healthcare is like people's crave now, and the business is another thing. So the major players, if you see, like you have Google. So these people have invested lots and lots of money into it, billions and billions of thing. So Google, Amazon, Microsoft, IBM. So all these people have invested, and it was around like $600 million in 2014, and it is predicted to be around 150 million billion dollar in 2026. So this drug industry is going to be a the healthcare industry is going to be a big boom. Involving billions and billions of dollars in the thing. So, so this is typically a healthcare system. Like you have a physician and a patient. So you have a medical tablet. So in future, like every physician will be having a system or a tablet in the sense of iPad kind of a thing, with which he is going to access his desktop. So all the information pertaining to the health. So now also our Prime Minister has announced this one nation, one health card scheme. So that means like all the health records or electronic health records will be stored in a cloud. So every patient details will be accessed with the help of a unique ID like your Aadhaar. So that will be stored in the cloud. So cloud is a public repository, public storage, wherein you store all the health records, and that will be accessed uh, from to your desk from your desktop, or to the, it will be given on the click of a thing to the physician. So that will be done. That will be like biobanks, clinical trials, wearables, EHR data. So all these things will be stored in the cloud, and it will be accessible to the patient as well as the physician. Okay, so that's a typical healthcare system that you will be seeing. So actually So these are some of the things like Google's healthcare. So Google is expanding its healthcare division to improve diagnostics and the clinicians' ability to treat patients. So it is plans to reinvent the 3 trillion US healthcare industry. Okay, so the CEO Sundar Pichai has said, like, so tomorrow if AI can shape healthcare, it has to work through the regulations of healthcare. So that is also very very important, because like when lot of data is being stored, the patient details, they're all very sensitive and private, isn't it? So the privacy and security is another major thing that people are working on, because like everybody should not be able to access the information of other patients. Only the patient and the doctor should be able to access. So the security and privacy plays a very role, and the regulations of healthcare industry has to be taken care of. In fact, I see that as one of the biggest areas is where the benefits will play out for the next 10 20 years is healthcare. So Sundar Pichai has told this. And Google reports that around 7% of its daily searches are health related. So everybody, uh, what kind of search they do. So 7% of its daily search, the Google search engine, are health related. And it receives around 1 billion health questions every day, or 70,000 health related searches each minute. See the volume of healthcare related search that is being done in Google. See how cautious or how quality conscious or healthcare conscious people have become. So Google, Microsoft, and Tencent are responsible for more than 70% of the deals made for digital health startups. And Google is number one with 57 digital health startups in its portfolio. So it has started several startups. So the company's investments are focused on genomics, clinical research, insurance, and benefits. So this is another thing that Google is partnering with the company verily to screen medical images. Say, for example, if a doctor has to screen all the patients, so it becomes so difficult for diabetic retinopathy and diabetic macular edema. So it has partnered with this verily installing the technology at a hospital in India, say Arvind Eye Hospital and all. In India, a shortage of more than 1 lakh eye doctors are there. So shortage is there. So only 6 million out of 72 million people with diabetes were screened for diabetic eye disease. So screening itself takes a big task. Like if you go to a ophthalmologist, like it takes hours together. You have to sit and wait for him because he has to screen one by one. So all these things could be automated. So many individuals go undiagnosed and untreated. So Google has invested a lot of money along with a sister company for screening this medical. So at the first stage, you'll be screened, and if you don't have any problem, automatically you will not visit the doctor. But if you have problem, then you can go in the next step and you can visit the doctor. So that is the kind of screening that you can do. So this is like screening of eye disease. Okay. And how it works is like patient is imaged by a trained staff using a fundus camera, and the image is uploaded to the screening algorithm through management software, and then automated machine learning screens for DR or retinopathy or macular edema, and the screening results are delivered immediately. So that is how it works. Then you have like Google Cloud EHR voice assistant called Suki. So I can show you a demo at the end of this if time permits. So it's a Suki is there. So that's an artificial intelligence voice enabled digital assistant. So it aims to help clinicians complete administrative tasks, including documentation or EHR information retrieval. So now the documentation has become a very very serious problem for the clinicians. Like they hardly have time to look at the patients to consult the patients, but in foreign countries it is expected that everybody has to fill this EHR, and that will be for insurance claiming and other reasons. So but India, like it is not that thing, but still in a tertiary care hospital like Jipmar and all, it becomes very very difficult. Per day they see around 400 500 patients, and how it is possible for a doctor to write very neatly all the clinical notes of the patient. So that becomes very difficult. So there if you have a speech assistant, like if the doctor keeps speaking and somebody translates it and keeps it, then it will be good. So physicians have reported that time spent on each note dropped 76% on average over the past year, from 13 minutes to 3 minutes, using this Suki. There is artificial intelligence, voice enabled artificial intelligence. So this is actually, this works with the help of deep learning models. So they developed to detect complication in chest radiographies. They did so with the same accuracy as radiologists also. So So this is the hands-free. So it says, Suki, create a follow-up note. Understand context without a Suki shows the patient problem list. If the doctor says like it will just give. So doctor need not search for the patient's reports. The next time the patient goes to the hospital, the doctor will not need to search for those things. So automatically it will search and it will give you. So just you have to speak and this Suki will take care of all the things. And these are some like artificial intelligence based chatbots. So why chatbots are nothing but they are kind of a robot. It's a software basically. It is available 24/7 and they are virtual agents and no need of appointment. So if you have some doubts like hi, I have this problem, so you can keep talking and that will reply to you based upon your question. It will answer. So how it will answer is it employs some kind of machine learning or deep learning techniques. So it will, because according to your question it should repeat again, no? So you would have seen that movie also, Endran and all those things, like their robotics, the Chitty robot. So there it is like it keeps talking to people according it answers. So it's all based on machine learning and deep learning. So you might not have a physical robot, but it is a software based robot. That's what is called as a chatbot. So it is available 24/7. So that is advantage. That is one advantage. And it's a virtual agent. So you can say that it's a virtual agent in the sense like you don't have a real agent, it's a virtual one. So it provides anonymity. Like sometimes people may feel shy to tell their disease to a doctor. So in this case it is easy, no? So they don't know the face, they need not look into his face and speak. So they tell their problems and accordingly they get the answers for them. So anonymity is there. People can talk without hesitation and no need of any kind of an appointment. So that is a chatbot. So there is other called Wobot. Wobot is like it's a mission to make mental health radically accessible. So this you can go and check up in https://wobot.io. So it is developed by some Stanford psychologists and AI experts, and people involved are storytellers, data scientists, engineers, designers, and so on. So people with mental health, say you're having depression with COVID, like many people went into depression. So if you're having depression, you need not go to a doctor. Like there is a social stigma, isn't it? If you go to a psychiatrist, people will say that you are mad or something. So always people are scared to go to a psychiatrist. Even for the slightest thing, they fear. So to avoid such things, like they can have this conversational chatbot. They can go to Obot and they can say like what kind of depressions they have and other things. So accordingly uh, it will give you some suggestion. Clear? So that's being developed by Stanford psychologists and AI experts. Okay. And so delivering cognitive behavior therapy to young adults with symptoms of depression and anxiety using a fully automated conversational agent. A randomized controlled trial is being done. So self-help program for college students who are in depression. So all these things we can. And Apple is using AI. How it uses AI is to screen children for autism. So researchers at Duke University, they have developed an app that uses the iPhone's front camera and facial recognition algorithms to screen children for autism. So that will actually help parents find out if the child is at risk for autism by showing a series of short videos. They will be showing some videos that elicit reactions in children, because certain videos will be funny or some serious. So depending upon that, whether the children are behaving, so that they want to find out. So in that case they the resulting facial patterns will be recognized, micro reactions and behaviors are analyzed in real time to give feedback if there is a potential risk for autism. So according to their lip movement, their smile, their eyeball movement and all those things will be captured by the app, and that will send a feedback to the parents, so to screen for autism. So that is one other thing, technique which uses AI. And then you have medical transcription. Amazon is investing a lot in medical transcription. I think you know what is medical transcription, so I'll not get into it. So this is the general perception. So AI, general perception is like AI is used to identify disease and so on, but not like that. Because machine learning or classification algorithms are used to identify malignant and benign cells for a long time. So identify normal persons from patients, and robots has been used for surgery. So this is a general perception that you have, but it is something more than that. A typical scenario on role of AI in healthcare. So if you see, like, it AI's role is omnipresent, like in all this stages. So patient visiting to the doctor or the hospital, the doctor may use a AI to screen him for some. So patients explains the problem and doctor uses a digital assistant for that. There you can have AI. So analyze the past medical history of patient. There you can employ AI. So it can scan, say if you have been a patient for a long time, you'll have bundles of old reports. So if the doctor will not be able to turn around all the pages and see like what has happened to you. But if AI is there, it will be checking all the complete history, and it will say like, okay, you have been diagnosed with this in a very precise summary. So that is one advantage. So analyze the past medical history of a patient and correlate with lab reports. So current lab reports in the past one, it can check. So because the doctor will not have time for doing all these things, he'll hardly spend 2 minutes for a patient. But the AI can do it in no time. And then speech to text conversion and store it in EHR. Inpatient analysis, like after admission, what are the different how he's improving, all those things analysis could be done. And continuous monitoring of patients by nurses and doctors. So for that also they can use AI technique for easing out their task of making repeated or mundane entries and so on. And then you have a prediction of a medical emergency, say like people in the ICUs can be continuously monitored for their vital signs or vital parameters, and whether there's a rise or a lowering of certain parameters, and accordingly it can raise an alert or it can give a notification or indication to the doctors through mail or WhatsApp or something like that. So you can do that. So this is a typical scenario. So you have AI in all walks of life, in each and every phase of a patient doctor encounter. Okay, so that is the thing that I want to emphasize here, because like it's not only dealing with disease identification or classification, it has got to do with so many things. So this is also a thing, like, okay. So key applications, if you see in healthcare, it is like disease diagnostics, classification, clustering, disease prediction, epidemics prediction, drug allergic symptoms. So these are on the right hand side, I just put like what technique you are using. So disease prediction means it will be doing a time series analysis, image processing. So this will be used for the computer science people, and the left hand portion is the application, and the right hand side is normally the technique that we use. So drug design, it's nothing but an optimization problem. Drug allergic symptoms, like you have clustering, holistic patient monitoring, correlation, hospital resource management. So these are all some of the application areas in healthcare. And these are some techniques, like okay, if time is less, I'll go directly to my drug design. So these are some of the techniques for machine learning: convolutional neural network, CNN, time series classification, classifying ECG and EEG signals for what to say, whether they are normal or abnormal activity. So for that they use a LSTM, which is again a machine learning technique or deep learning technique. And the image classification, the growth of tumor over a period of time can be captured as images. They might be subjected to several other factors. Normal growth and cancerous growth patterns could be labeled as time series data and used for further classification. So these are all the things that uh, do exist, but each one is a separate topic on its own. Like I can speak for hours together for each and every topic, but due to the paucity of time, I'll just go on to the drug design that we will be seeing. So as I told you that, so any questions till now? Shall I move forward? Okay. So artificial intelligence, like I told you, there are several sorry, expert system, neural networks, data mining, statistical AI. All these things are sub branches of AI. So here, what we have done, one of my PhD scholars, she was working on drug design, and we used this part. There is an evolutionary computation for that, and evolutionary computation, or we use the swarm intelligence. So both are coming under AI. So we use these techniques to design potential anti-diabetic or diabetic drugs. So we tried to do that. It was at a basic level, but what we call it as it's a multi-objective application. It's a drug design or drug or medicine design, because like we have a huge chemical search space. Like we have a number of chemicals, like in the form of acids and amines, and you need to solve multiple objectives in order to get a drug. So it's a multiple objectives to optimize. So actually drug design is a very complex process actually. So for a drug to I think all of you know, like for a drug to come into market, it takes so many years together, and it costs millions and millions of dollars. So that is a very very daunting task, and to do it in the wet lab, it is very very difficult. So to avoid that, they went for this CAD, that is computer aided drug design, or a CAD technique. So what it will do is it will take acid and amine fragments, and it will try to combine and do the testing, and that has to fulfill certain objectives. So there are something like Tanimoto similarity, Weber's rule, oral bioavailability, and so on. You people know more about it, but these are some of the objectives that needs to be fulfilled or to satisfy in order to get a drug design complete. So these are the multiple objectives which we wanted to optimize. So we used what is called as the swarm intelligence algorithms, which are inspired by the collective intelligence of swarm of birds, a school of fish, etc. And we use this bio-inspired algorithms could be used to solve the real-world optimization problems. So we which behavior we adopted is we adopted the biofilm life cycle. The bacterial biofilm life cycle algorithm was utilized, and this is basically the, so I cannot go in depth into it because it will take 1 hour minimum to tell about the complete thing. But this is an algorithm or a algorithm that simulates the biofilm life cycle to produce an optimization algorithm in computer science. Okay, so we utilized that, and what we did was like we took the drug fragment library. So it is already available, and there's a CDK package, that is a merged fragment molecules and descriptor values will be there, and reference molecules will be there. So reference molecules are, initially we took lidocaine and furanopyrimidine. So we wanted to generate some drug molecules which are similar to these two things, because that was a trial. We wanted to see like how similar we are able to get a drug by combining the existing molecular fragments or molecules. So for that we use this Mobifi algorithm. So that was the algorithm, that is our own algorithm, and we had given this Tanimoto similarity and oral bioavailability score to this algorithm, and that algorithm will be generating drug like molecules which are non-dominated solutions, in the sense which are superior solutions when compared to the others. So it will be generating a number of molecules, and we will be taking the good molecules from that. So this is the kind of thing, the fragments or molecules representation Mobifi. So we cannot directly put it in a container and mix everything. Like it is like we have to represent it. I told you, know, everything has to be represented in the form of numbers or numerical quantities. Accordingly, we have labeled those acid fragments and amine fragments. So acid and amine could be combined like this, as I have shown here, this red and blue. Or it could be it's a double amine fragment where you can combine acid fragments and amine fragment in between. It could be shown like this, and this is given to this algorithm, and you generate. So acid fragment 15 is this one, amine fragment 109. So likewise we are given some numbers. So phenotype is here, and this is the acid fragment, and so after combination it will give a drug like solutions like this. So we need to find out like which one is good depending upon various other factors. So that is the optimization, the role of the optimization algorithm that will do this. And what we thought was that after testing for this two, lidocaine and furanopyrimidine, we took other, we wanted to generate, we wanted to produce some anti-diabetic drugs which are similar to some herbal constituents. So we thought like anti-diabetic drugs are very useful. So we took some anti-diabetic herbal constituents like bitter gourd, jamun, this curry leaves, and this periwinkle flowers. So all these things, the chemical which is because see, because I am a novice to this actually, but we had taken all those things, because like bitter gourd and all those things, the chemical constituent which is present in that, which is creating insulin or whatever. So that component, that molecular structure will be given here, that is keratin, mahanibin, rutin, indolin and all those things. So we need to produce drug molecules which are similar to this. So for that we use the algorithm. So these are the things that we got. Routine like one, first one is the first component we molecule we got, drug like molecule. This is the second thing, routine like 2 molecule. And we try to compare with molecular descriptors, HPA, all those things we try to compare. So this was the things. The outer line shows the actual routine, but these are the other things that we have got, and what factors it is satisfying. So we try to obtain like this. And it was also evaluated, it was also evaluated based on a docking study was done, like because we don't know what is docking study actually, but we gave it to the bioinformatics department, and there we had a pharmaceutical, professor who is MPharm, PhD. So we gave it to him and he tested it for the docking. He did a docking study because we do not know, and he told like, okay, it is good. So docking score was obtained, and finally it was reported. So we just publish it in a thing, but it is only in the preliminary stage. You cannot directly use it for use it as a drug, because it has to be synthesized in the wet lab and then it has to be done. But the design of these things will be done, it can be done at a, because we got very limited access to all these libraries. Like if we are a very large pharma or something, then if we put our research into that, then we'll be able to make it as a full-fledged one. Since we don't have much access, it was a trial that we have done, and my PhD scholar had completed with anti-diabetic drug selling. So this was the research that was undertaken in our lab, and that is for your reference. So to conclude, like you have many hybrid models to support medical practitioners in diagnosis, treatment planning, and identifying risk factors to be developed. And we need to come out with some common formats for medical reports, because each record is different, and we need to find some common formats. And improved machine learning and deep learning techniques could be used. And finally, make modern medicine available to at reduced cost and waiting time. So Bill Gates has always said, like, we always overestimate the change that will occur in the next 2 years and underestimate the change that will occur in the next 10 years. So in the next 10 years, we cannot anticipate like what is going to happen. It is going to be a drastic change that is going to occur, and the machine learning and deep learning are going to sweep the healthcare industry and drug industry to a large extent. So that is about the talk. These are some of the references. I have not added much. Thank you.
Thank you, ma'am, for your nice presentation. So we have received some questions on chat box. We can see it.
I cannot see.
Yes. Minimize your PowerPoint, ma'am.
Yeah, that I have closed, but nothing is there in the chat box.
On your screen, you can see, ma'am, now.
I just posted hi, but
Now it is visible, ma'am? Not showing?
no, I'm not able to see the questions. Screen is visible. See, I just put hi, hello. But
Yes. Question is there on screen.
okay, what does AI mean for the skill requirements of health professionals? What does AI mean for the skill requirements of health professionals? Okay. So you mean that for health professionals, what is the AI component that has to be learned? Is it so?
Yes. This what are the
Yeah.
skills required?
What are the skills, like now, like everything has become open source. Like if you learn little bit of Python, that will actually help you to do some kind of image classification and other things. But like normally these are interdisciplinary fields. Like when you say, like, now the time is right for interdisciplinary research. So an AI expert cannot do everything on its own, or the pharmaceutical person cannot do everything on his or own, because you cannot completely learn this field, or I cannot completely learn your field. So it is time that we collaborate together and work on some projects, because like I will see to the AI part and you can see to the pharma part. But you should understand what I'm doing. I should understand what you're doing. It's mutual. So little bit of Python, little bit of programming. So this kind of lectures or this kind of seminars and what is AI. So those things will be enough. Like what could be done, what could not be done. If you understand that, that is sufficient. Or a little bit of programming in Python, it's good. Otherwise it's not possible to learn the complete deep learning and you start programming and we start doing pharmaceutical things. It's not possible. So it has to go on an interdisciplinary thing. Artificial intelligence can help to modify behaviors. What kind of behaviors, sir?
It is not mentioned anything. I think human behavior maybe.
Sir,
Any our activities, daily activities or any behavior of human beings.
you mean to say like voluntarily it will modify the behavior?
No, just our daily activities or any behaviors, ma'am.
Intentionally or unintentionally you want to change?
Yes, yes. We don't know. Some participant have given questions like this.
Yeah, because if the participant is there, like, okay, I can talk to him. I can say, like, what kind of behavior you want to change. Probably
No, yes,
you can detect the modification in your behavior. For example, you have
yes,
been
yes,
behaving in some way. You can detect
yes,
like how he has changed his behavior over a period of time. So for a period of
yes,
time, it will be observed. It's called as a time series data. So if you observe his behavior for a length of say 6 months, and suddenly he has changed, that could be observed and reported. But the machine cannot tell you, okay, do this or do that. It's still not there.
yes, yes.
Is AI useful in preliminary disease diagnosis? Yeah, definitely. That's the main reason AI has been roped in. The primary disease diagnosis, like people have been using AI extensively. Is it cost effective? Yeah, definitely is. It is cost effective because to get a doctor and he can see only this many number of patients per day. But okay, if you have an initial screening done by the AI, that will definitely reduce the cost. It is most cost effective and time effective. Minimizing. Is there any AI software for preclinical study? So what do you mean by preclinical studies, sir?
Ma'am, it is on animal study only. Whatever the drugs we are synthesizing or extracting from natural sources, we can apply on animals.
Yes. No, I'm
So,
not very sure because I don't know whether
yes,
there are any
yes.
specific software for this particular thing. But if you just Google and see, like for everything, like there are many free softwares or downloadable softwares that are available that you can just transfer.
So we can add here, ma'am. We can go for docking studies. So if docking study is showing any activities, then we can apply on animals.
Ah,
That
okay.
study can correlate. Yes,
Okay.
it will be useful. So docking study is one type of artificial intelligence. So we can apply that technique.
Okay.
Yes, yes. So
How can AI help address racial, gender, social disparities in healthcare systems? Yeah, definitely, sir. Like certain places, like it is not gender specific or race specific or socio-economic. Say people might not have money to get a good treatment. So now, as our prime minister said, like everybody should have a healthcare system in place. That is like all the details has to be stored. Say one person taking a treatment here, if they go say in Kanyakumari, if he's taking a treatment tomorrow, he's going to Kashmir. There also his health record will be accessible through the cloud, and the cloud from the cloud the data will be given to the doctor immediately. So it is not looking at any gender or race or socio-economic thing. So if it is going to be free, made free of cost to each and every Indian, then it is not going to take any of this things into account. So there is no race. Machine does not understand all these things. Is there going anything related to quantum computing to generate the drugs? Actually, quantum computing is in a very, very primitive stage. Okay, so quantum computing is going to sweep the world maybe after some time, because very few algorithms have been designed for quantum computing, and because the quantum computers have not come into force. Only if they come, then they apply that to drug industry. Is how can AI be useful to increase BCS classes? Say, what is this BCS? I do not know, sir.
I think some classifications of drugs for for different type of disorders. So
No, I'm not
I yes, yes, yes,
aware of.
yes, yes. So we can apply, ma'am, in different classes of drugs, like suppose anti-diabetic drugs or anti-hypertensive drugs. So different classes are there.
Yeah.
So he's asking that whether it is useful or not. So we
No, for
can
any
apply
drug you can
in a
apply, sir.
sense.
Unless and
Yes,
otherwise you
yes,
have the chemical fragment library, you can apply for whatever class of drug. That's not a problem.
yes,
It
yes,
all
yes.
depends on the availability of the chemical fragments. That is where your role plays a picture. Say if you contribute to the chemical library or the fragment library, then we can do our role.
Yes, yes.
Yes, sir. I have given the link in the slides. I'll share the slides, sir.
Okay, okay. Thank you.
Okay.
Thank you, ma'am, for your nice presentation, and thank you for accepting our invitations. So it will be very useful for the participants, ma'am. Thank you.
Thank you so much, sir. I'm really honored.
Yes, thank you.
Thank you so much, madam, for sharing your valuable knowledge with us. It's really gonna helpful to everyone in all aspects of their work. Here, I want to express my heartfelt gratitude for having you with us today. Now, I request to our beloved principal, Dr. V.V. Ravi Kumar sir, to present a memento to Dr. S. Sivasatya madam as a token of acknowledgement and admiration on behalf of the entire Roland family.
Thank you, Dr. Satya Madam, for sparing your valuable time and giving useful information about artificial intelligence and machine learning. I hope all the participants will definitely benefited by your lecture today. Thank you, Madam. And on behalf of Roland Institute of Pharmaceutical Sciences, I would like to present this E Certificate of Appreciation. Please accept it, Madam.
Thank you. Thank you, sir.
Thank you, Madam.
Thank you.
Okay, sir.
Okay, Madam.
I will take it. Thank you.
Uh, Now we will start our second session for this FTP shortly. For that, I request to Mr. Rajaram sir to continue the further sessions.
Yes, sir. Good afternoon. Sir, we are sorry for the late.
That's
Good
okay.
afternoon, sir.
Sir,
Good
Good afternoon,
afternoon.
sir.
Yeah,
you
I'm
can
just
continue,
figuring
sir.
out yeah.
Yes, sir. Yes.
Uh, okay.
Sir.
Hello,
Rajaram, sir, you
everyone.
can continue. Thank you, sir.
Good afternoon, dear participants, and welcome back to the session 2 of day 1 online FDP sponsored by ATAL. Dear participants, we are having with us our next resource person, Dr. Mohit Majumdar sir. Before starting his eminent talk, let me first of all introduce Dr. Majumdar sir to all of our participants. Dr. Mohit Majumdar sir is having over 12 years of research and industry experience. Dr. Majumdar sir has completed his masters in bioinformatics from Jamia Millia Ismailia and a PhD degree in computational biology and chemistry from Jawaharlal Nehru University. Sir is an expert in machine learning in application to biomedical and chemical data, having core expertise in structural biology, computational modeling of biological systems, and protein dynamics using long-range molecular dynamics simulations. Dr. Majumdar is a co-author in 28 international peer-reviewed research articles, 3 book chapters, and numerous international conferences. Dr. Majumdar has instrumental in developing bioinformatics facilities at the School of Life Sciences, JNU, and is a recipient of multiple awards, including the Indo-Sastri Award sponsored by Government of India, NVIDIA GPU Award by Fujitsu, and others. Sir is having 5 years of teaching experience working as teaching assistant in JNU, teaching bioinformatics to MSc and PhD candidates. Sir is currently heading the global business development for Pine Biotech USA and contributing to marketing and sales as well as mentoring participants from various programs, educational curriculum development, research project mentor, bioinformatics and data science course development. Dr. Mohit sir is contributing in the development of multi-university and company collaborations leading to more than 30 bioinformatics and data science training program and events in last 2 years for students in India, Africa and USA. With this brief introduction, once again I welcome Dr. Majumdar sir into this ATAL FDP program, and now I invite sir to deliver his talk. Over to
Sir, you are unmuted. Please unmute yourself, sir.
Am I audible now?
Yes, sir. Yes, sir.
Just a second.
Sir.
Hello, everyone. I'll just share my screen. 1 second, please.
କାଲି ପାଇଁ ମୁଁ ଆପଣଙ୍କୁ କହିବି।
Is my screen visible? Can anyone confirm?
No, sir. Just not came, sir. You go
It
to
is
share
visible?
screen option. No.
Yeah,
You go to share screen option. Then you select entire full screen.
Yeah, 1 second, please.
Sure.
Well, there is some permission issues. I'm just figuring out. I'm sorry for this. It's just the first time I'm using this, so maybe that's the issue. So just a while, I'll be getting this file.
Okay, sir.
So I'll have to join back again to make it effective, this changes.
Okay, sir, no issue.
So let me join back.
Yes, sir, just came.
Now it's visible?
Just a second, sir. Yes, make it full screen, sir.
Just a second. Actually, I think with the camera on, there is some issue. I'll just put my camera off and I'll go back to the presentation.
Okay, sir. Make it full screen
So
and
the
hide,
slides
sir.
are visible, right?
Yes, make it hide option. On lower side of your slide, make it hide. Okay. Okay,
Right.
nice, sir. Clear. Continue, sir.
I'm so sorry about this.
No,
So
thank you,
without
sir.
wasting much time, I would like to
Yes,
say hello to
sir.
everyone. And I welcome you all to this session on the overview of AI and drug discovery. So let me start by thanking Dr. M.B. Karvikar, sir, and Dr. S. Shiva Satya for the wonderful session on the application of artificial intelligence for healthcare and drug design. So it was great to gain the technical aspects of AI, and I would also like to thank other organizers, Srimati J. Jaya Lakshmi, Dr. BVV Ravi Kumar, and Dr. Vishwa Mohan Sahu. So today, in this wonderful faculty development program, we have already learned so much. I mean, we have learned a lot about AI, and to briefly say in this space, what we have seen so far is that AI can be used efficiently and effectively in different parts of drug discovery. So my topic is kind of very broad, and as Dr. Satya was saying, that each of these topics can be a separate topic on itself. So I'm going to do a brief overview where we will be discussing the application of artificial intelligence in drug discovery and why there is this need of artificial intelligence to begin with. So AI can be used in drug discovery, including drug design, chemical synthesis, drug screening, polypharmacology, and drug repurposing also. So there are several advantages of using the technology. However, I mean, there are some, despite its advantages, AI also faces some significant data challenges, such as scale, growth, diversity, and uncertainty of data. So in today's session, we will learn about introduction and application of AI in drug discovery and learn from industry examples as well. So, but before we do that and before we get there, I would like to first introduce our innovative company and our team of biomedical experts who have made this happen. So, to begin with, we are a US-based bioinformatics company who are working with multiple academic and commercial collaborators to develop easy-to-use analytical tools. And our mission is to make data science and bioinformatics and this AI and its application in biomedical and biological research more accessible. And we are doing this by providing a lot of training and tools that helps people make discoveries using this data. So we use next generation sequencing data that many of you might have heard of, and other type of high throughput data, which are very essential for this modern biomedical pipelines that have been adopted by pharmaceutical industry. And we will be learning more about that. So we see this as a result of this accelerated pace of biological data generation and availability of NGS technologies throughout different locations in the world. And there is a widespread appreciation for the accuracy and the detail of that data, of the such data that it provides us with. So, I mean, today I've been really honored to be part of this and to be part of this faculty development program, and having this opportunity to tell us about AI in drug discovery. But to tell you about the omics logic and how we've been able to do some of the things that I will go through in this talk. Omics Logic is a program for bioinformatics research and training that was developed between Pine Biotech and Tauber Bioinformatics Research Center. So it's a bioinformatics research center who are working to analyze big data, simplify it, and come up with interventions and collaborate with pharmaceutical industry as well as research institute. So what we will discuss today is the result of a hard work by an amazing team led by our company co-founders, Dr. Alfred Tauber and Dr. Leonard Brodsky. So Dr. Tauber comes from medical background, has expertise in oncology, immunology, biochemistry of inflammation, and he has a recognition of supporting this international grants and supporting these research projects where the initiative is to support and develop applications in medicine. Dr. Leonard Brodsky, he's the director of Tauber Bioinformatics Research Center at University of Haifa, and he's an expert of bioinformatics. He's an expert in bioinformatics and biostatistics, has numerous publications in mathematics, bioinformatics, and over his career has developed multiple novel algorithms that simplifies data and that uses artificial intelligence and machine learning tools. And the rest of the team includes Ilya Brodsky, who is our CEO. He's also a biomedical expert and is leading the organizational collaboration in the US. So together we have collaborated with UCSF and different universities to develop these research platforms. And the rest of the team includes bioinformatician, computer science, and training who are contributing in training. So to tell you about this, that over the course of past several years, we've been able to collaborate with several academic partners and been doing evaluation of curriculums. So bioinformatics and data science curriculum and how AI can be applied to some of the things that leads to project design. And these omics logic program and training were then thoroughly evaluated. That led to excellent outcomes throughout the year. And we've been working in several locations as well. So today, the Omics Logic International Program is being running in 7 different regions and have 10,000 people enrolled. So that includes like a rich community of researchers, faculty, biomedical data scientists, biologists, and clinicians. So together, there was a question about what we can contribute. So as a biologist, as a clinician, as a student, we all are contributing into this because the requirement is like that. And that is due to the fast growth of data, and that we will be going to understand in the next few slides, where we will be discussing about that how everyone can contribute and become a citizen scientist at a student level and become a researcher or a PhD level research to do and work on these technologies and come up with something which is which would be really a boon for healthcare system. So our team is working with local and regional coordinators for this, because it's just not possible. And I mean, with universities and with faculties, they are helping us to refine these things, the curriculum and online training, and adapting them to the needs of the students and researchers. So that was about the company, and about myself. Already have had such a elaborated introduction, and thank you so much for that. So my name is Mohit Mazumdar. I have finished my PhD from Jawaharlal Nehru University, and during that time, I was able to publish 25 research articles during the PhD time, contributing in bioinformatics analysis and prediction, and that helped in taking decisions for wet lab experiments. So these contributions led to recognition that helped me develop grants for more research and travel, and then went to EMBL Sanger Institute in the UK and UFS in Canada to work on drug screening and identification. So being someone who have finished a PhD in data-driven era and moved to industry, I thought it would be worth sharing my experience. So after finishing my PhD, I joined a bioinformatics company, working with big pharma research institutes to help them with their drug discovery pipeline. So there I learned that what is the actual need, and what is what we actually learn throughout our career, throughout our academic career. So I could differentiate that how where the application is currently, what is happening in the industry, what is happening in research, and where we are doing in education, and how much of that we are learning. So after gaining that experience about the need of the industry for data-driven research, I joined Pine Biotech. And here, the purpose of this is, in my current role, as said, I'm collaborating and I'm working with students, faculty, and scientists. And we are providing support to supplement existing bioinformatics and biotechnology coursework at various institutes, universities, colleges, and develop a bioinformatics ecosystem, to be said. So along with our hardworking and efficient team, we've been able to work with more than 30 universities in India and helped over 1,000 students to understand how they can implement data-driven research and come up with research projects that make sense for all of the hard work that all of us are doing, to be able to come up well, and to be able to get recognition, or to be able to contribute. Now, coming back to today's topic, how AI, drug discovery, data science, all of this is making sense now. So the pharmaceutical industry is undergoing a dramatic shift. And as said in the previous session, that this shift is so dramatic, it's very difficult to predict what would be the next thing. We have seen these things developed over time. It did not happen just now. So why now? I mean, the reason is this gradual increase in information. So information has been been growing ever since in 1990s. In 1900s, where we started recording these experiments. We started keeping information about this, and for a century, there has been contribution from so many people. I mean, only if I talk about India, we have published so many articles that have contributed in the data growth. Similarly, all around the world, US, China, Europe, the data has been published and deposited from all over the world, and that led to a concept of big data, which now helped with the emergence of advent of artificial intelligence and machine learning. So while many are quick to paint this new technology also as an imminent threat, so it may instead could be a solution also to our chronic drug shortages that we are seeing. So in fact, AI has already been successfully applied in various facets of drug discovery and development, from helping scientists find new potential medicines to predict which drugs will fail in clinical trials. So this all require a lot of data, because as said, machines machines, if you think about machines, machines are good in recording a lot of information, and they are also good in remembering that information. So the more information we give them and the more structured information we give them, the better the AI is. It's as simple as that. So as more pharma companies beginning to adopt these technologies, there is no question that they will significantly affect the future of the medicine. So what exactly does this mean? So to be able to understand the use of AI, we need to first learn about the big data in biology, because the big data in biology is the increase in the information about different types of data types. They include highly detailed, high throughput data obtained from sequencing experiments, as well as screening experiments, assays. So now on the screen, you see some of the applications and analysis that played a critical role in modern drug discovery pipeline and development of AI models. So genomics, transcriptomics, and understanding of the structure, DNA level and information, protein level information, cellular level information, tissue level information, and if a systematic system level information. Then at a morphological level, topological level, sequence level, and the profiling, I mean, how do you segregate them into different categories? Then there are interactions at a molecular level. Then at a genetic level, you have options to find signatures. So all of this is being together makes complex interactions, and that complex interactions leads to complex regulation of subcellular biological mechanisms. Then when we have a disease situation, we have to identify key mutations and variants. So you have heard about all the variants that are coming up, right? Delta, alpha. So that is for a virus, and that's a small genome. And then we have situations in cancer where we have human genomes to compare with. So those are some of the things that are the application, and where AI and machine learning are playing a key role. So understanding about the molecular mechanisms of mechanics of drug designing first, and then by understanding that information, how do machine learning there also plays a role to generate these new molecules, to come up with new molecules and assist with the discovery part. So this is some of the things that are there. I mean, they look all a lot of a different research field altogether, but they in combination. Now they are grouped together into this field of data science, AI, and another very important field that has come to an age is bioinformatics, because as it involves the analysis of biological data, right? So what is bioinformatics? So bioinformatics, it is a convergence of biology, data, where both are equally important, as we will see in the next few slides. So who uses bioinformatics? Who is it for? The question might have been, the answer might have been for, first, it could be statisticians, it could be computer scientists, it could be engineers. But the truth is that, I mean, more and more biologists and clinicians are now expected to understand and handle their own data, to publish and succeed in both basic and translational sciences in today's world. So in fact, biology itself is rapidly acquiring the characteristics of data science. And why so? Because as the technology improved, we saw that there's a lot of automation in every level of experiments that we are doing. May that be talking about these NGS experiments, or may that be structural biology, crystallography. Even in crystallography now we have robotics to address to set up the experiments. So data generation is kind of becoming more and more, I mean, easier with all those with advantages of getting all those technology involved in it. So now data getting the data is easier, but now analysis of the data is the biggest challenge. And that's why biology in itself, life science in itself, is going to be data driven. So let me start today by mentioning that as a company in this space, we are focused on addressing some of the pressing needs in biotechnology and clinical markets, which by working with researchers and clinicians to leverage transforming opportunities. And so this is the time for this is a time for opportunities, and this is a time for our lifetimes, I guess. So big data, which we have heard about for past several decades, is absolutely transforming the healthcare system, making it more dynamic, most dynamic. And I know that many of these technologies and in the impact and the data is being generated in the West. So obviously we still are doing our best and going there slowly, where we will have a system soon to understand and get most of the information together. So but I would say that we are approaching this opportunity from a very different perspective as well. As a company, the starting stage of research and development in drug discovery, I mean, talking about the drug discovery market and the big data market, you have to understand that why to adopt for this. So in general, I mean, starting from research and development phase, R&D phase in drug discovery process, it itself takes around 6 years. Sorry. Whereas the next phase for clinical trials, on average, takes more than 5 years. So just after that, just 10 out of 10,000 initially tested candidate for new drugs makes it to the clinical trials during that time. So we have 10 molecules. So by and large, at the end of this lengthy drug design process, just one medical product out of every 10 is what that enters the actual clinical trials, which goes into the process of approval, could be used for patients after getting approval by the regulators. So according to the US FDA, approximately 33% of the drugs which moves from phase 1 to phase 3, and then around 25 to 30% are the drugs who move from phase 3 to the next phase. So there's a lot of issues there because of the cost and time. So all of that. So many companies in this space are focused on data quality, aggregation, in a way, collection, effectively processing, and sophisticated analysis. So these are the areas to focus on because, as I said, like if there are companies who are working on these areas, there are job opportunities for the same as well. So these are indeed important components for a successful transformation. However, we need to remember that the center of in the center of the story are the people. So AI, drug discovery, machine learning, they are as good as the team. I believe in that, and as good as the data. So we think that everything could be automated and everything could be rationalized and the way we vision it or imagine things. But in reality, when you look at data and the information, the metadata, all the information which is lacking, it's really a tough job still, because we were never thought of that, or we were never careful in doing the reporting properly. So drug development is a costly, time-consuming, and a gradual process that begins with identification of a small molecule and ends with final new molecule entity, right? So the main aim is to find an active particle where the ultimate goal is to affect the body. It will affect the human body and prove that it has the quality, purity, and usefulness in treating patients, right? So the foregoing criteria that guarantees that the endorsements by the regulators, new medications, and patients' quality of life, not only by treating their condition, but also by ensuring that the drug does not become a source of other issues like adverse reactions. So there are a lot of challenges in this, and how data is going to play a role in this is something where the focus is and where the opportunity is. So overall, with FDA report, the final, from starting to the counter, reaching to the counter of the drug cost around 20 million. And you heard about, like, maybe it's an inflated number, but you heard about the revenue that are the big pharmas that they generate. So I think it's a problem, because then the innovation is only centered for those few big farmers, and not for others who could also play a part in this innovation. And because it affects all of us. So if the process is so difficult that only those who are super rich farmers can be able to do it, then it's a big problem, and that has been a problem. So this, to reduce this enormous, spending investment in today's world, scientific progress is data driven, and that's why we are adopting to data, because it gets us the answers. But also there are challenges, as I said. It is because of the structured or unstructured data. So how bioinformatics and biology as data science. So besides we discussed about costs, we discussed about regulatory barriers that becomes a concern, leading to high failure rate in selecting new drugs. So this is critical, because I mean, the logic of the analysis and the training that you go through for learning about biology as data science starts from the point where you're doing the data generation. So you are generating more and better data, and for that you need to get insight about the data. So what is the problem? There is a gap. So what happens in a regular system is that if you are dependent on someone to do the data analysis and understand and makes sense of your data, then you may sometimes lose some information there. So having some insights and gaining some understanding about how you can explore your data which you have generated is very important. And then for obviously specialized analysis, we are there's a model to collaborate, and that's the best model. I think ma'am also recommended that. So yeah, obviously. And then making decisions out of that analysis, and then try to go in that direction. So if I have instead of screening thousands of molecules, if I can screen billions of molecules using deep learning, and if I come up with a solution with one molecule, or with five, if I come up with solution for 500 molecules that are like really, I mean, are going to be effective, that actually saves a lot of cost. So that's one of the reasons that this has moved to the field of data science. So importantly, I mean, today there is a growing appreciation. So that you can see in the context of data generation, computational method for data organization and analysis, and as well as visualization. So I talked about exploratory analysis in the last slide, and exploratory analysis is that in AI, what we talk about is data sets. We discuss data sets in all the aspects of business, whether that is a business data. So similarly in biology, it's also data sets. If AI in healthcare is anything is about, it's about the data sets. So the field that enables us to generate data and to understand about those technologies that are generating that data is an important factor, and then it's a key to be able to work and contribute and do something in this, to be able to understand the technologies that are generating this sort of data. So what technologies? I discussed today some of the technologies that are there helping us understand cancer research really well. So how has it been possible? So let's understand this in a little bit more detail, because one of the important aspect of being able to get an overview is to be able to come up and understand some of the basic factors, some of the basic data types that are involved in the pipeline of drug discovery for pharmaceutical industry or for biomedical research or for all the labs that are working around the world to be able to come up with this. So one of the reason is more usage. Obviously, you saw that it was very expensive back then, and based on the usage now, by institutes, governments, countries are now investing to develop infrastructure for genomic testing, right? That is leading to lower and lower cost of conducting this high throughput assays or analysis. So I think it would get more and more affordable as we progress further. So what is the data that we are talking about here? Just a second. And if I'm missing out on any questions, I'll look at them. I'll look at them once we take a break here. So yeah, so I was telling you about the technology. So first we have to learn about the technology itself. So the technology, which is the next generation sequencing technology, is one of the technologies to get information at a sequence level, right? To connect the sequence level information, genetic level information to the phenomics. I mean, to the features that we see. Why someone is tall, why someone has a special characteristics. So all of those questions can be answered from this genetic level information. So let's look at some of those technologies that are helping us to use and understand about us, ourselves, and the surroundings and all the living creatures. So omics technology like NGS can be used to explore the roles, relationships, and actions of various types of molecules that makes up the cell of an organism, right? So this sort of information, many of this omics data can be generated using next generation sequencing, or what is known as also high throughput sequencing. So this data can show detailed information about genetic variants, pattern of genes, histone modification and non-genetic changes, and then also about the isoform expression and understanding of whole protein expression at a particular time point. So these are the experiments that are helping us develop models in AI so that we can predict some of the disease ahead of time by looking at biomarkers. So before going to the application of the technology, which is also a part of this today's session, I would like to tell you about these individual technologies. For example, genomics. You must have heard about genomics because it's an essential field, and it's also an essential subfield for bioinformatics. And it's a major force in expanding human knowledge of genetic association with disease and other traits. So it's an interdisciplinary field, including the development of methods for DNA sequencing, as well as for big data analysis of genomic sequences. So next generation sequencing techniques allows us to look at the whole genome sequencing and analysis of epigenetic factors, also I said, such as DNA protein interaction, DNA methylation, and the information that we get is at an unprecedented efficiency. So massive data sets for biological sequence data such as GenBank, EMBL, SRA, and NCBI. So one of the good advantage of the research, the way it is that to get grants and publications, we do have to submit this data in a public repository that makes this data available to all of us. And it's an opportunity as well. So data-driven research and knowledge discovery. So that is about genomics, and there are some applications, there are some techniques like what we do is comparison basically. So we have a disease state, we have different haplotypes. So we kind of do alignment and see if there are changes, then understanding the significance of those changes and annotating those changes and all of that. This is something that we do often in our company, working on the platform. But I mean, in general, genomics is a very important field, and almost all of the bioinformaticians out there are some way working on some aspects of genomic research, which is also helping pharmaceutical companies and also other research labs who are working on AI and analysis of large data sets of mutations to be able to predict those mutations are also contributing. Another field is metagenomics. I don't know if you have heard about it, or maybe all of you have heard about it. It's a very exciting field of research. So metagenomics is the study of collection of genetic material, genomes, from a mixed community of organisms. So metagenomics usually refer to the study of microbial community, and it's also referred to as field. So genetic characterizations of organisms which are too small to visually identify also requires computational methods to make determination of how and where differential gene expression within a microbiome contributes in cooperativity or permissive ecosystem, and then the effect. For example, we have had examples about the gut-brain axis, how the gut microbiome affects our health. That's a key area of research, to give you an example. So this information provides a framework for studying sequence homology, performing taxonomical classification effectively, and then also the concept of classical microbiology at a genomic or an organism level genomics. So this is something which is this field is really literally used in with viruses a lot, and there's a case study that I would be going through in today's session, which would be about COVID-19. Another field is transcriptomics. Transcriptomics is a study of transcriptome, where the whole set of RNA transcripts that are produced by genome under specific circumstances. So there's a time point, obviously, where you collect the sample, and also there may be a disease state, or there could be different things for this sort of research, or in like specific cell, single cell transcriptome. So using high throughput methods, collecting data for gene expression. Now, where all of this data is, like as a researcher, as a faculty, if I want to do analysis and I know that there is a lot of information, but what to do with that information? How do we where to find it, and how do we process or move next? So NCBI gives a lot of information about these sequences, and I saw yesterday, maybe they are also going to introduce some of the cloud technologies which they will be offering to analyze the data. But there are so many data sets that are available already on different aspects of disease or translational or basic research, which is there to be seen by the researchers, the PhDs, or the researchers, or the lecturers, or the faculty, to be able to come up with research questions that could help the society. So I think it's a great resource which could be utilized by all of us and contribute. So if I have to conclude, then I would say that when existing dots are paired, when you connect the dots, I should say, perhaps, great breakthroughs happen. So penicillin, for example, got its start in 1928, when Alexander Fleming returned from vacation to find a mold has infected some of his bacteria cultures, preventing the natural growth of the bacteria. However, it wasn't until a decade later that Howard Foray came across Fleming's article on the penicillin mold in the medical journal. Then it gained traction. So with his colleague Ernst Chain, he started discovering penicillin, and his first human patient was successfully treated with the drug in 1942. So from 1928 to 1942. So a sequence of dot interactions results in one of the most significant medical breakthroughs of our day. So connecting the dots is very important, because a lot of information is out there. That's what I wanted to say. And this is similar to how artificial intelligence and machine learning are used to link data dots, to connect data dots to yield new insights. So once we get information about a single individual, for example, if you have the genomic information, if you have the transcriptomic information, if you have other gut information, if you have epigenetic information, think about collecting that and integrating that. So that is something is what is, integration of data and connecting the dots. And why I'm saying connecting the dots, because sometimes some of the answers that are unexplained from one technology, and we have been using one technology, one sequence, one protein for a long time in our research. So I'm saying that as a researcher, and now when we have the information, high throughput information about various technologies and various types of data, it's time to integrate that data to find solutions. So that integration, and there is complexity in that. So the complexity is that that how to do it, because there are so many things that we need to learn about or we need to know about, is it not? So machine learning is becoming an essential technology for drug discovery and development. AI platform architectures can process vast amount of data, which help researchers in discovery and development of drugs by providing them with valuable insights. So at the same time, it is important to understand the technicalities of each approach to make the right decision for your machine learning projects. That's very important. That there is a way to do this. The way to do this is to do the literature review. So there's so much information which is out there. Connecting the dots, you can only connect the dots when you have the dots put together. So the dots needed to be put together, the information need to be structured, and your research question has to be finalized. So if all of that information you have, and if you have an idea about it, then it's a matter of applying, and that is not such a big task. The important thing is to collect information and come up with the question that could be answered with that data. So that's the first phase where we discussed about the overview of AI in drug discovery. Next phase, I have a few more slides, and if I don't know if we can continue, because I'm already in the lunch time. So maybe we can do this on a different time, or I'm looking for suggestions. Should I go for the complete slide deck, or should we conclude here?
Continue. Continue, no problem. Am I audible,
Dr.
sir?
Mohan, yeah. Sorry, Dr. Bishwa Mohan, sir, can you hear me?
15 minutes, no problem.
I couldn't hear you. What did you say?
Sir, you can continue next 10 minutes also, no problem.
Okay, thank you. I'll go
Yes,
back to my
sir.
presentation then.
Yes, sir. Yes, sir. Yes.
So the next thing that let me know if you guys are getting bored or anything. So I'll just, or have any question, you can tell me that. So next part of the thing that we did is some example of application of artificial intelligence in drug discovery. So application of data science in biology. So many computer-oriented strategies and other methods were used to invent and optimize lead compounds, right? We talked about today molecular docking, pharmacophore modeling, selecting using random forest, then comparative during molecular dynamic simulations, sampling a lot of structure at a time point, quantum mechanics. So quantum mechanics in understanding of the charge distribution of the system and generating a highly specific simulation is a possibility for a maybe smaller protein. And with Google, with their prediction software that came very recently, they were able to said that they were able to predict the problem of protein folding, right? So there has been a lot of things that have been happening in this space, and that has led to these big companies to create their own platforms. So Google have their own platform, Nvidia have their own platform, we have our own platform. So the technology that you develop gives you an opportunity to come up with your own company. Maybe that's an entrepreneurship option. And also to be able to apply this. I'm putting more emphasis on applying this, because there is a lot of tools and techniques and algorithms and which from other fields you can adopt. So there is already there. So many people are more interested in developing something new, like an algorithm, working on something, and thinks that is only AI. But applying AI to the problems is something which is of immediate need. So collecting data, applying these methods and technologies, and finding solutions is one of the place where we should all focus on in general. That would help us. So what I'm going to talk next is about one of the application of genomics in SARS-CoV-2. So with Biomed, Biomed is an organization in the US. We have partnered with them. It's a new, so in that we are analyzing the sequencing data. And so let's take a look at that. So we know that the SARS-CoV-2 pandemic has had a severe consequences in everything that we have been doing so far. It has affected all of us, in simple terms, badly, yeah, of course. But SARS-CoV-2 pandemic has changed also or transformed our appreciation for collecting this information. So the appreciation of genomics, for example, now India have a lot of labs who are doing the sequencing. And then once you have the sequencing data information with you, there's a lot of bioinformatics that you need to do. So understanding how genomic data analysis tools can help identify specific viral strains, and then understanding via multiple sequence alignment that how multiple genomes, if you align them, gives what sort of information. So next is also phylogenetic analysis, where you understand about the evolutionary aspect and the significance of the mutation in the context of viral protein structure and function. So finding that key mutation and then understanding that what will be the effect of that mutation. It's something that we all are interested in COVID-19 also. So we will further discuss here about the data-driven process. So why bioinformatics? So together we will understand how data analysis tool can help detect viral genomes in next generation sequencing data. So that is one of the key objectives of this program with LPRN. And then identify the specific haplotypes from the NGS files like FASTA or FASTQ. So these are the data files, text files that you get from the analysis. Then do the comparative analysis that I was discussing a little while ago to gain insights into vaccine design. So you see the changes, find the changes that are happened, and look into the principles of antiviral drug design. So just not like have a structure, have a mutation, shall we go for drug designing? No, there are other principles that you have to look into while you're doing drug designing or while you're doing vaccine designing. That includes the identification or characterization of the entire structure, the amino acids, and the development of, many times what has happened is like with those Q mutations, there's a development of resistance to standard drugs, like, so that multi-drug resistance leading to superbugs that does not respond to standard antibiotics, we can find solution to that based on the genomic data if we have a population data. So the application of this is being used to understand. So there are some key objectives like data generation, data processing, quality control, sequence analysis of the viral genomes, because where this data is collected from. So I'm going to tell you about that. So the data is being collected from wastewater. So analysis of phylogenetic relationships, I said, of the strain, also the population, also the host. Analysis of variants at a level of function, at a level of structure, and also the associations. And then structural analysis of the variant. So this is a typical variant analysis pipeline. So we kind of build these pipelines to assist biologists so that they don't have to do the installation and they can run their big data experiments on the cloud. So this is just a screenshot from there. So these bioinformatics pipelines and workflows are quite essential in the analysis of genomics data generated by NGS. So using the analytics and the coding, a little bit of R and Python coding, which is also there on the platform, helps us to develop and rich library and tools that are available in Bioconductor and GitHub. So processing this sort of data and then understanding that what this data means. So bioinformatics methods that find individual mutations like multiple sequence alignments can be combined with evolutionary models that track changes in the genomic sequences and link them to a process. So finding those mutations in the context of the population and use them to track. So these models fit to thousands of genomes that reveal this dynamic tree that you see here. This is an open source tool, and I will be pasting the link for you to see this after the presentation. So you will be able to see that how mutations have diverged and at different locations. So next train is the website. You can also Google that. Now, all of this information what is being the effect. So in early February, US laboratories were only sequencing about 8,000 COVID-19 strains per week. Since then, the rate of sequencing has increased substantially, strengthening the country's ability to detect and respond to emerging of these contagious COVID-19 strains. So like the variants currently sweeping through the Midwest and the past, they have been swept the Midwest and East Coast. So the Biden administration, which came, has already made 200 million investment to help increase genomic sequence to 29,000 per week. So wastewater sequencing and how do we do this? So the process was to collect the sample from wastewater treatment plants and then centrifuge the sample and get that RNA, and then quantify RNA and determine viron numbers. So after this, utilizing the pipelines for processing and analyzing of genomic data, that our team was able to study genomic variations that in the collected samples and identify commonly reported mutations in some from the samples. So from the wastewater to getting the information on computers with the mutation. It sounds like very, when I say it's in 2 sentences, but it's a very trivial task. So we got encouraged by the preliminary results. So the collaborative team now is seeking to join funding for expand the number of samples so that the LSU, Louisiana State University, which we are collaborated with, and potentially the whole city to benefit from the advance notice if a novel strain emerges. So if there is a new strain in the waste and it comes to the wastewater, obviously all of the human wastes are coming through there. And there are different regions it is coming through. There's a segregation. So you can understand and pinpoint that this is the location where this is so this is genomic monitoring. So this is one of the applications. So what we are doing is that we are preparing tutorials and resources, and we also run this as a program where you can create a report to analyze variants using genomic sequence data, so using cloud processing and R script. So this is the kind of information that you get at different amino acid substitution points, and there's a whole tutorial course and all of that for this. So this was the case study. So you saw that there's a genomic monitoring happening in real time, and in that we are facilitating the genomic analysis, but it's efficient system to be able to apply this at the very beginning. So now artificial neural network and deep learning, because the lot of data, a lot of information. So how ANN and DL. So in most and almost every science and technical field, deep learning algorithms are seen as one of the forefront spaces of research and development, right? We know that you've been hearing about that. Deep learning algorithms enable computational models to memorize and master a representation of multi-dimensional information through abstraction. So multi-dimensional information, not a single. So deep learning requires a lot of big data sets, huge number of data sets compared to machine learning. So deep learning also solves many problems faced by that are being faced by machine learning algorithms. So for example, image analysis, speech recognition, these are two one of the most used fields. In the process of drug development, the deep learning method has been used as an exemplary method for predicting drug activity, also targeting and discovering basic new drug molecules and new drug candidates. So one of the example I would wanted to give is about precision medicine, and Benevolent AI is a UK-based company. They actively develop drugs for diseases such as ALS. So ALS is amyotrophic lateral sclerosis, and then Parkinson diseases, then UC, ulcerative colitis, and sarcopenia. So what they do is that they have established alliances with big pharma companies, which are helping them or funding them to do this for themselves, for the big pharma. And then they have the AI drug discovery platform, where you can do cutting edge and experimental to find new kind of molecules. So to find new kind of molecules, they before that use the integration of data, omics data, and then for drug targeting, for finding the target molecule, and then they have these small molecule libraries, and they create dynamic libraries also from using graph theories, and there are some other machine learning techniques. Similarly, biopharma has this biomarker discovery, which is just recently we just talked about drug discovery, and we talked about biomarker drug discovery. So biomarker drug discovery, one of the major challenge is clinical data is very noisy. So that is what I wanted to point out from this, that the challenge is also about the kind of data that you get that is very noisy. So here there are techniques that are used so our team worked with the clinical center to evaluate their measurement system of a disease to help select responders to a combination of treatment. So it's like application of precision medicine. And then there is small molecule screening. So the vast chemical room, which contains thousands of molecules, encourage us to go for see the changes. So in shortage of the modern technology, on the other hand, that inhibits the drug discovery process, AI is playing a key role because in identification of hit and lead molecules. So drug optimization, drug target identification, all of that is included in this. Then we have the complete application. I would wanted to describe a paper here, but I think I'm running out of time, and Just to tell you that the most innovative biopharma organizations are currently exploring different models to harness the AI-powered drug discovery technologies by striking several. They are doing this by also, gaining access to small companies that have come up with the innovation. So yeah, that's there is a lot many collaborations from big pharma where you will see recently in 2 years where they have acquired the AI based companies just to be able to work on all these signatures and able to work on bigger data sets. So with this, I think what we can do as a researcher is to be able to apply this technology and data sets by doing some of the means of processing, understanding the formats, applying big data, extracting meaningful insights, and then applying it for different type of clinical application. So patient stratification, disease classification, diagnostics, identification of potential responders to the therapy, mechanism of disease onsets and progression. And then there are biomarker discovery in pharma, detection of toxicology, and then analysis of molecular mechanism for drug discovery and drug repurposing and repositioning. So as I said, these are research areas that we can talk about all day. So to work with the faculties, what we have been doing also is to be able to work on some of the projects. Some of the projects are listed here. So this is a faculty student group collaborative grant proposal we are working on. There's another project, Dr. Prabhuda and their students, and then there are student groups working on multi-omics approach on student faculty groups on fish and also like agriculture. So we have tomato ripening, understanding their ripening and non-ripening, then tardigrades. So all of this are some of the examples of application the students can exactly do this. So I think I would want to finish this with these some examples, and I hope that today's this presentation was informative for all of you. If you have any questions, I'll be happy to help you out with the answering those questions, and you can email me at mohit@pine.bio. And I'd like to really thank you all for giving me this opportunity to talk about this important topic. And I would like to thank you all for listening.
Thank you, sir, for your nice presentation. So it will be very useful for the current scenario in the research. So I think all participants will be benefited by your presentation. Thank you, sir. So we have not received any question on chat box. I think they understood all part of your presentation.
Thank you.
Sir.
Yeah, you can email me also. I mean, if anyone have any queries.
Yes, sir. Yes, sir. Yes, sir. Rajaram, sir, proceed.
Okay, sir. Thank you so much, Dr. Majumdar, sir, for sparing your valuable time for all of us and sharing your research experience and vast knowledge with all of us. We remain most grateful to you, sir, for the rapt attention with which you have delivered this presentation. May I now request our principal and convener of this ATAL FDP, Professor B.V.P. Ravi Kumar, sir, to present an e-certificate of appreciation to Dr. Mohit Majumdar, sir, as a token of our acknowledgement and deep sense of gratitude from entire RITS fraternity.
Thank you, Dr. Mohit, for giving an overview of the advancement of artificial intelligence in drug discovery. It will be very useful for all of us. And on behalf of Roland Institute of Pharmaceutical Sciences, I would like to present this e-certificate of appreciation. Please accept it, sir.
Thank you, sir. Thank you so much I really appreciate it. Thank you.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Thank you so much, sir. Dear participants, With this, the second scientific session of day 1 has come to an end. We will be resuming the third session very soon. Good afternoon, dear participants, and welcome back to the session 3 of day 1 online FDP sponsored by ATAL. Dear participants, we are now having our next resource person, Dr. Balakumar Chandrasekaran sir with us. Before starting his eminent talk, let me first of all introduce Dr. Chandrasekaran sir to all of our participants. Dr. Balakumar Chandrasekaran sir is currently working as an assistant professor in the Faculty of Pharmacy, Philadelphia University, Jordan. His area of specialization and research is pharmaceutical and medicinal chemistry. Dr. Chandrasekaran sir has graduated the B Pharmacy degree in 2002 with first class from the Tamil Nadu Dr. MGR Medical University. After qualifying GATE 2003 exam, sir has did his M.Pharm in pharmaceutical chemistry from LM College of Pharmacy, Gujarat University, India. With AICTE fellowship, sir has graduated his M.Pharm degree with distinction in the year 2005. After serving as a lecturer in two different pharmacy colleges for about 2 years in Gujarat, sir has joined for PhD in pharmaceutical sciences at University Institute of Pharmaceutical Sciences, Punjab University, Chandigarh, with prestigious research fellowship in Science for Meritorious Student Award granted by UGC New Delhi. After his PhD work, Sir has joined as an assistant professor in Karpagam College of Pharmacy, Coimbatore, India in the year 2011 and promoted to associate professor and HOD Pharmaceutical Chemistry Division in the year 2013 to 15. Sir has supervised 5 M.Pharm projects and backed few scientific research publications. Sir has made significant contributions to the Karpagam institutions by serving in different capacities as Dean in charge at Karpagam University for facilitation of PhD projects, member Institutional Human Ethical Committee, Karpagam Medical College, and team member in establishing Karpagam Pharmacy LLP, which is presently running over 100 different formulations. In the year 2015, Dr. Chandrasekaran joined as postdoctoral researcher to conduct research in the development of novel anti-cancer drugs with the fellowship from School of Pharmacy, College of Health Sciences, University of KwaZulu-Natal, South Africa. In the year 2016, Sir backed the internationally prestigious fellowship from Department of Science and Technology, National Research Foundation, South Africa, to continue the anti-cancer research project for further 2 years at UKZN. Dr. Balakumaran has supervised PG students and established various international collaborations with countries such as USA, UK, France, Germany, Czech Republic, IACT India, and Saudi Arabia. Consequently, published several research papers in very high impact journals. His area of interest includes computer-aided design and development of novel anti-cancer agents, anti-tubercular agents, anti-inflammatory agents, antimicrobial agents, and anti-asthmatic agents. Dr. Chandrasekaran has got 2 international patents to his credit, and he is one of the editorial members of Elsevier, Willis, and different frontier journals. With this brief introduction, once again I welcome Dr. Chandrasekaran sir on board to share his valuable talk with us. Namaste sir.
Thank you, Professor Rajaram, for your nice introduction. First of all, I say my special thanks to convener of the faculty development program, co-convener of this faculty development program, and the coordinator, Professor Biswa Mohan, for coordinating this very wonderful ATAL. Initially, I say that ATAL, I thought some name, but AICT sponsored training and learning projects. So it is very good initiative in the Roland Institute of Pharmaceutical Sciences. So I wish your team to be successful in conducting this online faculty development program. So I say thanks to all of you for giving me this opportunity to present the integrating aspect. So what we say that as an academic person, we work in drug design, which is computer-aided drug design, and pharmaceutical compound synthesis. So we say this is the integrating approach. We apply computer-aided drug design technology into the pharmaceutical chemistry to facilitate the drug discovery projects. So I just to interlink between these two with the major aspect, that is artificial intelligence. Nowadays, it is a hot topic. So I can I start my presentation?
Yes, sir. Yes, sir.
Yes. Thank you. Thank you.
Go for share screen.
Yes. Yes.
Yes, yes.
Is it visible now?
It not still not came, sir. Go for share screen, then select full screen mode. Yes, yes. Enter screen mode.
Just a minute.
Yes, yes. Just came. Just came. Make it full screen, sir. Make it full screen. Go to enter screen option, sir.
Share screen.
Then inside under the entire screen, select that one.
Share. Entire screen share. Okay.
Yes, yes.
Can you
Yes,
see
yes,
this
just-
slide? So now.
Yes, yes. Make it full screen now.
Yeah. Is it okay? Now.
Yes. Then you hide, sir. Yes, hide the slide near lower side.
Hide.
Just
Hide
make
the
it
slide.
Yes,
Okay.
yes. Just click.
Is it okay now?
Yes, yes.
Is it fine now?
Yes.
Okay.
Yes,
Okay. Good afternoon to all the participants and faculty members. I am going to present artificial intelligence in drug design and synthesis. So as we see the earlier presentation, it covers entire aspect of artificial intelligent applications in drug discovery. So in drug discovery is a wide application part. In that, the initial stage of drug discovery is drug design. So I'm going to cover the significance of artificial intelligence in drug design as well as chemical synthesis. The question, why do we the new drug discovery process, then little bit about artificial intelligence, then artificial intelligence in new drug discovery, artificial intelligence in drug design, in synthesis, and speeding up the drug discovery process using artificial intelligence programs. And also going to discuss about various challenges in drug discovery under artificial intelligence applications and future problems and possible solutions. So why do we require new drugs? The question starts, the answer could be to treat infectious diseases due to evolution of new microbes and new variants of the microbes. For example, COVID-19. And nowadays, there are mutant strains of COVID-19. So to treat these new evoluted viruses or any pathogens, we need to find new drugs. Similarly, to treat emerging drug-resistant microbes, such as, say, for example, drug-resistant bacterial strains, such as methicillin-resistant Staph aureus, vancomycin-resistant Enterococcus. So these are the drug-resistant bacterial strains. Similarly, the drug-resistant mycobacterial strains. So Mycobacterium tuberculosis, as prolonged administration of drugs, it can develop resistance towards multi-drug regimens. Maybe sometimes it may lead to extreme drug resistance or total, if we can say that total drug resistant MTB, which is resistant to all kind of drugs. So in that situation, we need to find new drugs. Similarly, secondary infections, such as now you can see the secondary fungal infections like black fungus, white fungus. So these are emerging. So they also need to find new drugs. Though we have the current amphotericin or azole-based drugs, but we need to find new drugs to tackle possible secondary infections. Similarly, to overcome side effects or toxicity of available drugs, to treat few neglected tropical diseases, such as African trypanosomiasis, Buruli ulcer, Guinea diseases. So for these purposes, we need to find new drugs. Now, we are going to discuss the entire new drug discovery process. So, what are the stages in new drug discovery process? So, if you want to invent a new drug, it has to undergo various stages to reach into the market. So, it approximately takes 15 to 20 years and $1 billion of investments. So to cut short the lengthy process, the computer-aided drug design is very much helpful. So I just highlight the steps involved in new drug discovery process. Start from target identification. So what do you mean by target? Target means the drug finally reaches to the particular protein. We say that it is a target. For example, receptors, enzymes, say for like DNA. So they are called as targets. First, we need to identify potential target from the disease mechanism. Then we need to validate the target. Target validation means to make sure the selected biological target is a key target, say that a key point of enzyme or key receptor. Once we validate the target, we can identify the chemical structure which is going to inhibit that particular target, or maybe the drug, we can say, or we can say a lead. So we need to identify a lead to bind to the particular validated target. Once we identify lead, it may have some physicochemical problems, or there may be some toxicity parameters. So we need to modify the structure, the chemical structure of the lead molecule, so that it can confer the druggability or drug likeliness. From that, we will find out particular, optimize the structure. So we will find the optimized structure. We will make a in chemical laboratory. We will synthesize those compounds. After synthesis, it will go to in vitro evaluation and in vivo evaluation. In vivo evaluation using animal models, so pharmacology, in vivo animal pharmacology. Once the drug qualify and it identified its efficacy, it will enter into the human study. We say that clinical trials. So clinical trials usually 4 phases, but 3 phases are very important. Phase 1, that is evaluation of new drug involving healthy human volunteers. Whatever the new drug we have identified, we will administer to the healthy people and estimate the parameters. Then it will go to the phase 2 clinical trial. This is nothing but a drug evaluation involving small number of patients. Once it is passing through the phase 2 stage, it will enter into the phase 3 clinical trials. In this drug evaluation, takes a large number of patients in different countries, as well as under different races or under different conditions. So once the drug passed through the phase 3 stage of clinical trial, it can be approved by the approval agencies or regulatory authorities to make the drug available into the market. So now the drug is available to the market. This is the new drug. Patients are administered this new drug. Then if there is any side effect or toxicity, it can be reported. So this reporting is known as post-marketing survey. So it has to cross through several stages, and each stage is time consuming, and it consumes a lot of money. So this new drug discovery process is a lengthy, tedious, and very challenging. So how to minimize the time factor or even cost factor to a certain extent, we have the drug design. So drug design is the early stage of drug discovery. So the drug design nowadays taken with the help of computers, we say computer aided drug design, CAD. Okay, so in this we have 2 types of tools. One is structure based drug design. The second one is ligand based drug design. What do you mean by structure or target? So as we say that protein target, what we discuss is the protein target. It may be a receptor, it may be an enzyme, it may be a DNA. So we need to find the suitable key protein, and we need to know the active site or binding site in that key protein. So we take those information, and based on the active site of the protein, we design the structure of new drug. This is called structure based drug design. So usually the computational tool involved in this is the docking or the dynamics. On the right hand side, we have ligand based drug design. So in ligand based drug design, we usually do not have the protein structure. What we have is chemical structure of the standard drugs, chemical structures of the available drugs. We take information from those chemical structures of the standard drugs, and we will calculate properties, and we will identify what are the significant properties which is contributing to the binding, to their binding with target proteins. So we will consider the properties. So based on that, we design a new drug. We will compute properties of the new drugs. So this methodology is called ligand based drug design, LBDD. So usually 3D QSR and pharmacophore modeling they are known as ligand-based drug design. So whatever the drug design methodology we use, we will come into a final form is lead structure. Once the lead structure will undergo in vivo, in vitro pharmacological studies, that is preclinical studies, later on it will reach to the clinical studies. Finally, it becomes a drug structure. So these are the two technologies, structure-based drug design or ligand-based drug design, finally reach into the point called lead molecule. So lead will become a drug after in vivo, after in vitro, in vivo pharmacological studies and clinical trials. So we just see structure-based drug design. So as we discussed, the target protein structure is required. The mechanism may be well established. The ligands of the structure, ligands that is binding to the binding site of the protein may be known or it may be unknown also, no issues. So we can perform a structure-based drug design methodology. So example, molecular docking is a very good example for structure-based drug design. On the right hand side, ligand-based drug design, here we will have information about the chemical structure of the standard drugs or drugs which is acting on particular target, but we do not know the structure of the target. We know the active drug structure going to bind to the target. So we calculate properties. We consider different three-dimensional properties. All this information we consider. Based on that properties, we derive a equation, and we on the parallel time we will identify the potential chemical structure from drug data bank or maybe some different databases, and we calculate the properties and we will substitute into the standard equation. We will estimate the expected biological activity. So this is called ligand-based drug design. Okay, so these two are very important tool in drug design strategy. So when we say that here in silico tasks in drug design, so we have some of the companies, they will integrate both the technology, means ligand-based drug design, structure-based drug design, both can be integrated for the drug discovery projects. So how they will integrate? First they will start from pharmacophore modeling. So they will take the standard drug structures and derive a pharmacophore model, and then they will screen the large drug data bank structures to the validated pharmacophore model, and finally and they will take parallelly they will perform structure-based drug design such as molecular docking, molecular dynamics, and come out a better lead structure. Then lead will be optimized, then it will become a drug. So nowadays companies they are using integrated technology. So start with ligand-based drug design based on the available standard drug structure, and continue with structure-based method to reach into the stage of drug in the market. If the condition is, if the protein structure is available, then only it is possible to perform structure-based drug design. So if protein structure not available, directly from ligand-based drug design, they will reach the drug structure. Okay. Now we will come to the point artificial intelligence. The definition, it is a technology-based system involving various advanced tools and networks that can mimic human intelligence. So when we talk about the intelligence, memory, learning, whatever we do, the teaching, cognition, everything connected to the brain. So we can say artificial intelligence is an artificial brain network, or we can say neural network. So artificial intelligence utilizes systems and softwares that can interpret and learn from the input data to make independent decisions for accomplishing particular task. Say for example, we want to find drug likeliness compounds with good pharmacokinetic properties. So this is our decision. Our decision is to identify druggable structure. So we can use this artificial intelligence based calculations. So those utilizes mostly softwares and neural network programming. So it involves several method domains like reasoning, knowledge representation, solution search, machine learning. Okay, we will come to the machine learning one by one. So subfield of machine learning is deep learning. So artificial intelligence is nothing but generally it is a machine learning technology. So if you take a machine learning, go to the deeper learning method, we say that deep learning technology. We say that deep learning tool, which is based on artificial neural network. So we will see the history. Actually, it is started 70 years ago. 1950, it is started, artificial intelligence, the terminology. Then it reaches to the computers in medicine. In 1975, the National Institute of Health sponsored a workshop on artificial intelligence and machine learning. Then in the year 20 years ago, deep learning was introduced and it is applied to the technology 2007, and 2010 it is applied to the endoscopy. We say that robotic system. So we say when we do the surgery for especially for the surgery as well as the disease diagnosis, they will insert a tube, micro tube. Say for example, if you want to visualize inner part of the stomach to find certain ulceration or the area of ulcer, if we want to confirm, then they will do the endoscopy. So in that it is camera, with camera it is inserted, and the inner part will be visualized very clearly on the computer monitor. Then 6 years ago, pharma boat is developed. What is that farm boat? We do the drugstore. In drugstore, so you see the robotic system is introduced. So when the pharmacy shelf is there, so this robotic system will correctly pick up the particular the drug package from particular shelf, and it will deliver to the pharmacist. So pharmacist will easily take the medicine and he will distribute to the patient, and he can spend more time drug in the self, in the pharmaceutical system for the dispensing of the drug and taking correct packets from the particular self. Okay, so cloud-based deep learning was introduced for the drug design projects 4 years ago, 2017. And recently, so we say that gastroenterology, certain companies are discovered new drugs based on the artificial intelligence programs. Okay, so this is about history. Then artificial intelligence classified in different ways. Say for example, based on the application, it is classified: reasoning and problem solving, knowledge representation, planning and social intelligence, perception. So perception, it is important in our drug discovery projects, as well as the machine learning we know. And robotic motion manipulation, it is very much used in the clinical side, polypharmacological side, pharmacological side. And natural language processing, it is also clinical and to improve the patient's cognitive factors. So we will see one by one. In healthcare industry, as we see that it is very useful in research wearable drug discovery projects. Patient data, such a huge data is stored and analyzed, and it will be delivered to the user. Similarly, for hospital management, surgical, and even it is helpful to the normal lifestyle. So come to the main point, machine learning. So machine learning calculations are three types: supervised, unsupervised, reinforcement learning. And supervised, we have regression. So in the statistical models, regression model and classification model. And these are very much helpful in the QSAR, QSPR. So quantitative structure activity relationship, that is QSAR, QSPR, quantitative structure property relationship. So to calculate, to decide various properties, physicochemical properties, also the formulation properties, we can optimize it. So that is statistical based method, regression analysis. Then unsupervised learning, we have two classes, that is clustering, feature finding. And reinforcement learning, we have decision making. So to decide the final decision of the drug structure. So for this, all these three type of classes coming under single umbrella, that is machine learning. If you go into the deep, it will reach to the deep learning methodology. So deep learning, it is a type of processing and also for decision making purposes. So it comprises maximum of neural networks. So we'll see the definition. These comprise a set of interconnected, sophisticated computing elements involving perceptions. So it is like a neural network. So perception analogs to the human biological neuron. So it mimics the transmission of electrical impulses in the human brain. So we say that machine learning, so three types it is helpful: artificial neural network, decision making, and instance based algorithm. Here what we look importantly, the perception, the neural network. And on another side, we have support vector machine, k nearest neighbor. So they are very much useful in QSR studies, quantitative structure activity relationship studies. K nearest neighbor, it is earlier used 1970s, 80s. And self-organizing map, the map will be generated based on the input data, and it will show the region to improve the activity, and it will show the region which is unfavorable region in a drug structure, which will detrimental to the activity. So these are all these part are very much applicable to the QSR side. And here we have the on the other side, artificial neural network. It is applicable to the both structure based drug design methodology as well as ligand based method. So mostly it is helpful in the structure based drug design methodology. So recurrent neural network, convoluted neural network. So these are the three important networks very much useful in target protein structural methods. So the companies which are working is International Business Machine and Watson supercomputer. So they suggest treatment strategies for cancer and also identification of the cancers, diagnosis. Okay, next, artificial intelligence in drug discovery. It is starting, it is helpful in various stages of drug discovery, starting from drug design. So in drug design, we have various drug databases: Zinc database, PubChem, binding database, NCI database for anti-cancer drug discovery. So from those databases, chemical structure, we will look properties. We will calculate the properties, physicochemical properties. So the chemical structure will be the chemical structure will be identified in its SMILES formula. So each chemical structure can be written in a formula which is acceptable by the computer, which is called simplified molecular input line entry system, that is SMILE format. So computer can recognize the SMILE format instead of the structural format. So using those different formats, we will sort out the suitable structure by filtering method. What is that filtering method? We will filter the particular drug structure based on drug likeness parameter, ADME, that is pharmacokinetic parameters, pains. What do you mean by pains? That is pan-assay interfering substances. So certain chemical structure we can say obviously bind to the most of the targets. So we say that pan-assay interfering substances. So we say the pains. So those type of compounds to be filtered, and any compound which is toxicity, they are filtered. So for this filtration process, the machine learning technology is very much useful. So the machine learning technology, we have all kind of techniques, neural network, then support vector machine clustering, all these things. So finally we lead to the QSAR or pharmacophore modeling. This is ligand based drug design methodology. And if we take information from the protein structure, then it will implicate to the target based drug design, such as molecular docking and molecular dynamics. So whatever the technique we use, we will come we can sort out the suitable structure which is having drug like property. So that we can identify, then it will be finally energy calculated. So we say that any drug structure binding to the protein which should be minimum possible energy, okay, and it should have better pharmacodynamic parameter. So based on those parameters, it will be sorted out and it will be prepared and it will be tested pharmacologically. So these are the various point of application areas in drug discovery involving artificial intelligence technique. Okay. So on the other side, I just explained how the classical method becomes modern method. So classical QSR equation, classical QSR model is nothing but we derive a equation based on the known standard structures. We derive a equation. So that is based on the classical methods, linear methods, statistical methods. We use it. Nowadays we are using modern method by integrating artificial intelligence tools. Okay, so neural networks, it was there earlier, but nowadays these are showing promising results. So artificial neural network is very much helpful in QSR, QSPR, and structure based modeling method, de novo design, and chemical synthesis as well. So in chemical synthesis, the recurrent neural network method is very much useful, and rule free prediction all are very much useful. Okay, so we have highlighted important points of application in artificial intelligence. And what I am going to talk in this presentation is artificial intelligence in drug design and drug synthesis. So in drug design, it is useful to predict target protein, to identify which is the possible target for treating the disease. Then later predict target drug interaction. So what if the drug structure binds to the protein target, what kind of interactions it is showing. Then finally predict the drug structure activity before synthesizing it. Okay, so these are the three significant stages artificial intelligence are applied. Next, come to the point of synthesis. To synthesize the drug structure, it will design a synthetic route. Synthetic route can be designed based on the application of these neural network programmings, and we can predict the possible reaction mechanism. According to the reaction mechanism, we can control the yield, or we can calculate the expected yield. Before going to the chemical synthesis, we can predict all these parameters. Similarly, drug repurposing. So predict a new target for the existing drug. So as we say that COVID-19 earlier stages, anti-malarial drug chloroquine was proposed. So chloroquine is already existing drug, which is purposefully used for malarial treatment. So nowadays they are repurposing to the COVID-19 treatment. So how they identified? So based on this application of artificial intelligence tools into the field of drug repurposing, then predict new use of the same drug or entirely design a new drug. So this is about drug repurposing. Coming to the pharmacological aspect, we can predict the activity. Similarly here in the drug design also, we can predict the toxicity, predict physicochemical properties. Okay, we will come to one by one. Artificial intelligence in drug design and synthesis. So what I say here is design, make, test, analyze. So we can design a new structure or identify suitable druggable structure from the drug data bank. So we say that design aspect. So in design aspect, these three important tasks are performed. QSR based drug design, that is ligand based drug design, and parallelly ADME property, that is drug absorption, distribution, metabolism, elimination, and toxicological property prediction, and design of new molecular structure and virtual docking. For all these things, it is helpful. Then after design, it is very much helpful in the synthesis. So we can design a synthetic route based on the input drug reactions. So synthesizability estimation, synthetic route selection, and reaction conditions. Or is there any possibility of side product formation in the synthetic step, or how the synthetic route is efficient, how to optimize the better synthetic condition? For all these things, this is artificial intelligence is helpful. Of course, structural elucidation, biological assay testing, then analyzing the data. So 4 step which is going as cyclic process. We say that informatics in drug discovery cycle. So design, making, testing, analyzing. So coming to the point 1, artificial intelligence in drug design. So in drug design, as we discussed, 3 stages it is helpful. Predict the target protein. So we can identify suitable drug target structures. So different softwares are helpful. In that, very useful softwares are: number 1, AlphaFold. It is based on deep neural network calculations. It is used to analyze distance between adjacent amino acids and the corresponding angles of the peptide bonds. The second software, Potential Net, it is also based on neural network. It is used to predict binding affinity of the ligand. Ligand means the chemical structure. Okay. Third one, Delta Vina, it is a scoring function for rescoring drug ligand binding affinity. So these are the softwares, and we take a simple example of what are the various stages it is helpful. Okay, so this is the conventional method. So what we take, suppose we want to model a particular protein from the template protein. Say for example, we can take a very good example. I say that cyclooxygenase enzyme we take COX-1. So COX-1 structure we take it as a template. Based on this COX-1 structure, we design or we can model COX-2 structure, another protein. We say comparative modeling. So all these are done by done with the help of softwares. So these are the conventional method. How we do the protein modeling? Take the template sequence structure and identify the suitable fragments and optimize the fragments, build a model, refine a model, analyze a model, estimate quality of the comparative or homology model. So this is the traditional method. Sometimes it can be integrated to certain statistical parameter and we make a integrated model. So we say that we can integrate along with the simulated models. So we say that sample and simulating, clustering, atom based refinement, all these things. So these are the conventional methods. Now what we say here, artificial intelligence, we take two important calculations. So number one calculation here, recurrent geometrical network. So this calculation will helpful to predict particular protein structure. In this also, what they are going to do, protein sequences are fed one residue at a time to the computational units of recurrent geometric network. So the structure is converted into fragment, and fragment is analyzed and their torsional angle. So angle on the dihedral plane. So all the torsional angle were computed. Similarly, the chemical structure is taken and their torsional angle or bond extension, bond rotation, stretching, they are calculated. Then it will be combined to get a whole structure, entire structure. Okay, then this is predicted structure. If protein data bank have already deposited target structure, then it can be compared and find the RMST values. From that, we can say that our predicted structure is how close it is with a structure deposited in the protein data bank. So based on these computations, torsional angles, their structures were predicted, and RMST is used to measure deviation of the predicted protein structure with experimental structure. So that RMST value will give the differences. So if the RMST is not deviating too much, it is very close, then we can consider that our predicted protein structure is valid and it can be used for further analysis such as docking or molecular dynamic simulations. Okay, so this way we can predict the target protein structure. So this is point one, protein structure prediction. Then point number two, drug protein interactions. So we know that when the drug structure, that is chemical structure, binds to particular protein or the active site or the binding site of receptor, it will interact with binding site amino acids. Okay, so we need to predict the interaction between the chemical structure and the active site amino acid. So for that, two important calculations are useful: random forest and support vector machine. So these are the workflow. From drug bank database, drugs and targets were identified. Then from that, drug properties are separated and protein target properties are separated. So they are known as descriptors. So it is converted into mathematical model and it will be made into the pool of database. From that, two sets were sorted out: training set and test set. Training, from the training set, we will develop a model and we will test the model using test set, and we validate the model and use, and it can be used for the prediction of drug protein interaction. So this is the flow chart. Next, coming to the one method, we will take an example of random forest. So in using the random forest, what the experiment says, drug bank database were taken and descriptors, dragon, profit, web server, they are used to find the descriptors, and random forest algorithm. What do you mean by algorithm? It is nothing but rule-based calculations. So we say the name algorithm. So random forest algorithm is used and derived parameter such as sensitivity of the model, specificity of the model, compound protein pairs, and area under curve. So bipartite graph learning. So it is applicable to the all type of protein targets such as enzymes, receptors. Receptors, we have four types of receptors. We will see in the next slides. So similarly, it is useful to predict top 5 scoring novel drug target interactions. So it can identify the identify the target and identify the interaction between the target and the drug. So it will give the scoring value, binding scoring. Based on that, which drug is better binding to particular target, we can identify. So in this way, drug protein targets can be, drug protein interactions can be identified. Similarly, novel target predictions for existing drug. So MDMA, methylene dioxymethamphetamine. So this is the psychoactive drug. So for this drug, what are the possible protein targets? So we take a drug, we try to find possible proteins where this drug is going to bind. So using this technology, top 5 scoring targets were identified for the same structure, same drug. Okay, beta 1 receptor, carbonic anhydrase 2 receptor, alpha 2A adrenergic receptor, prostaglandin enzyme or peptidase enzyme. So these are the possible targets for the same drug. Okay, so target prediction. Similarly, for the same target, what are the different ligands going to bind? That can be predicted. Okay, so we take an example, thymidine kinase enzyme. So we apply to the artificial intelligent programming. So top 10 scoring ligands for the same target. So that can be predicted and binding score values are given. So we can choose, okay, for thymidine kinase, if we prepare a drug that is nicotinamide adenine dihydrogenate, so this can bind very well, efficiently, okay, or edoxuridine. So this is also another agonist that can bind to the thymidine kinase. So we can predict possible drug structures that is going to bind to the same target. Okay. Next one. Yes, what we say that it is helpful to all type of receptors. Say for example, GPCR receptors, cellular networking is very much useful, and another for this iDrug program is useful for GPCR drug interaction predictions. iDrug CHI, it is useful for predicting drug interaction between drug and ion channel receptor. iDrug enzyme, so it is used for the prediction of enzyme or enzyme connected proteins. And iDrug NR, it is useful for the prediction of the drug protein interaction between a drug and nuclear receptor as a protein. Okay. Yes. Next, the third point, prediction of pharmacological activity or affinity of the drugs. So deep neural net QSR, it is a Python based tools. So Python is a program which is used in the homology modeling or any other computational tasks. Okay, so using those Python based program, that is deep neural net QSR. Neural graph fingerprinting is used to predict properties of novel molecules. Deep tox, it is used to predict toxicity of the drugs. Deep DTA, PADME, wide DTA, deep affinity. They are some of the deep learning methodology useful for the prediction. And we take one example, PADME. So a protein and drug molecule interaction prediction. This program is called PADME. It is useful. It is based on the interaction strength, how strong the drug can bind to the active site of the protein. So it takes consideration of both compound and protein information as input. And one of the study by Feng et al., they have used molecular graph convolution method for compound featureization with protein descriptors. They are also used multiple cross validation for the validation of the PADME program. So this is successfully applicable to the androgen receptor. So how they have used to the androgen receptor as a protein, and they identified that androgen receptor interacting with different type of drug structure. So they have used two programs, PADME ECFP, that is PADME extended connectivity fingerprinting, and PADME graph convoluted architecture. So these are the two programs, two methodology they have used. So in that SMILES format of the chemical structure and protein sequence information of the protein structure, they are fed into the architecture that is called either ECFP or it may be graph convoluted method. Any method they have used, and they finally identified the interaction strength. So if interaction strength is good, it can proceed for further development. So they took an androgen receptor. They have collected the ligand structure, that is chemical structure, 1 lakh structure from the NCI database, that is National Cancer Institute database. They have taken 1 lakh structures against the same receptor, androgen receptor. They have used both the methods, and they have predicted the binding strength and average of what they did. They have integrated both the method and find out the average of the prediction as average method. Out of 1 lakh structure, they have sorted out top 30 structure, they are binding strongly. Out of top 30 structure, they finally sorted out 4 chemical structures as druggable and very active. So these are the structure. So they have given input structure 1 lakh, output they got 30. Out of 30, 4 structures are highly active and druggable property. They exhibit druggable properties. So this is the natural ligand, androgen steroid binding to the androgen receptor. So androgen receptor involved in the progression of cancer, all these diseases. Okay. So by this way, this interaction strength can be predicted. So thereby drug protein interaction were established. So this is the drug, standard drug, this is the protein. From there, they have predicted new structure, interaction strength of new structures from the database, NCI database. So we say that. Okay. Now come to the second part of my presentation, artificial intelligence in synthesis of drugs. So different softwares are useful: computer, deep chem, organic. So these are the various software tools used for the chemical synthesis of identified structure, chemical synthesis of identified active structure. So regularly used softwares in the industries are Chematica and IC Synth. So we talk about in the synthesis 3 important points to be considered. One is retrosynthesis, reaction conditions, and reaction outcome. Okay, so 3 important stages in the drug synthesis. And when we apply to the artificial intelligence in deciding the synthetic route, synthetic reaction conditions, and output. Okay, so what we decide? First, we design the chemical structure of the highly possibly active drug structure. Now we need to make it in the laboratory. Before making into the laboratory, we want to consider economical aspect and the time aspect. If we want to decrease the cost and time, we will use this artificial intelligence tool. Say, for example, we have identified this is the target chemical structure, or this is the drug structure we have identified using drug design methodology. Now we want to synthesize it. Okay. How to design a synthetic route? Earlier we have manually, based on the literature and based on the reactions, reactive centers, we design a synthetic route. But what the computer will do, computer, as we say that it is a neural network. So neural network from the brain, central nervous system, we say in physiology, central nervous system, brain. From brain, they are rooted at the nerves. Nerves are separating and it reaches all over the body. Same way, now we want to fix this structure. We want to find the synthetic route for this target structure. How we will find? So we do the retrosynthetic aspect. What do you mean by retrosynthesis? Retrosynthesis means reversal of actual synthesis. What is actual synthesis? We take one reactant A, and we do combine with the reactant B, and we get product C. This is called synthesis. Now what is retrosynthesis? Means opposite. From the product structure, we will make a fragmentation and we reach the possible reactants. So when we say this is the product, so we make a fragmentation, or fragmentation means the possible synthetic pathway or synthetic route. Okay, from the fragmentation we will find for each fragment how to synthesize this fragment. That is based on the further breakage, further fragmentation into the small building block. Okay, these are the small building block. If we say the peptide synthesis, single amino acid is a building block. Same way, we from the product structure we fragment this point or this point or this point. So we make each fragmentation and we see whether the fragment selected available or not. Based on the availability and the cost factor, we will decide the fragment. Based on the fragment decision, we can able to synthesize the total molecule, total drug. Okay. So for deciding this synthetic route, reaction condition, reaction product outcome, so artificial intelligence tools are very much useful. So when we talk about the retrosynthesis, reaction conditions such as the use of catalyst, use of reagent, use of solvents, temperature or pressure or reaction time, all can be optimized easily. So it can save our time and cost, or cost of the chemical can be saved. Okay, we can save our time and money. Okay, then we can also predict the percentage yield. Suppose if I select this particular building block, what will be my yield? Suppose if I select this particular building block, what is my yield? Similarly, the cost factor. So $10 it is taking. So instead of going this route, I can go for this particular route. So likewise, it is helpful. Okay, this is called CASP, Computer Assisted Synthesis Planning, for synthesis of drugs. So point number one, retrosynthesis. It is a reversal of actual synthesis. In retrosynthesis, 3 points are important. One is single step retrosynthetic pathway and multi-step synthetic pathway. Once we decide a pathway, then we need to find the we need to find the reaction condition. If condition is optimized, then we can move forward particular reaction into the synthetic laboratory. Okay, so retrosynthesis, can be broken into sub problems of generation of retrosynthetic route for one step at a time, then use of recursive tree methodology. We can identify, optimize single step process. From the single step synthesis, we can go for multi-step routes. Okay, multi-step synthesis. So this is retrosynthesis. Retrosynthesis involves 3 stages. So designing of the route, designing of the condition, deciding of the reaction prediction. Okay, reaction conditions and reaction predictions. So here, expert encoded rules. There are 2 types of artificial intelligence are applicable. That is first wave artificial intelligence, second wave artificial intelligence. First wave is crafted based on the crafted knowledge. Second wave artificial intelligence based on the statistical methods. So 2 categories of methods for scoring compounds, such as simplified structure-based heuristics or full retrosynthetic tree expansion. So 2 categories of methods are followed. They first need to identify reaction centers based on possibility of changing of atoms. Second point, identify atoms near to the reaction center, then add general functional groups that is involved in the reaction step. Similarly, a single step retrosynthetic recommender is sufficient to construct routes for one step reaction. Okay, then it is extended to the multi-step reaction. Each step can produce thousands of precursors. Okay, we will see how the software flow will work. So IC-Synth, it is the normal software developed by the Infochem company. So they have this is the application tool. So you can see we can feed the parameters, algorithm parameter, and suggestion how many steps, what is the medium or connection disconnection approach. We can say retrosynthesis based on the disconnecting approach for synthesizing the target drug structure. Okay, IC-Synth software. So basically it will give the tree based algorithms, and we can find out the suitable synthetic routes based on the recommendation. So based on the possible 63 synthetic routes, we can sort out based on 2 things. One is economy and second is availability of the building block. Then only we can synthesize in the organic laboratory. Similarly, another method we have ASCO software. So say for example, this particular drug structure, it can be synthesized in 3 steps: acylation, deprotection, borylation. So if we want to make 1, 2, 3, 4 derivatives, we want to make 4 derivatives. So first unsubstituted derivative can be planned. Okay, can be planned based on the cost parameter. Suppose if we go for the compound number 8, 9, 10, so they can be synthesized using another extra step, 4th step. Okay, 1 step we need to move forward, that is N-methylation. And if we want to make the final structure, we need to go for 1 more step, that is Bernoulli reaction. Okay, so from that, if we want to decide synthetic route for this, we need to find the disconnecting point or the building block. So these are the disconnecting point for this. These are the building blocks, and the building block cost we need to consider. Based on the cost factor and based on the availability, we can go for actual laboratory synthesis. So before that, we can predict using this IC-Synth software, which uses DNN, deep neural network. Second, we know the reaction condition, how much quantity of reactant to be used, volume, how much ml, and how much concentration. Can I use catalyst 1% or 2% or 1 mole percent or like that? Then reaction time, 1 hour I need to do reflux or 2 hour or 3 hour, like that. Then order of adding reagent. Catalyst can be added along with the reactant, or catalyst can be added later point. So all kind of suggestion we can get from the software. So we can plan a reaction condition. Once we plan a reaction condition, say we take an example. Suppose it is a drug structure. we can go for two possible ways. Conventionally, we can go Suzuki cross coupling reaction when the diboron part, here we have the connecting point. So this, how to make a carbon-carbon double bond through this removal of the leaving group based on the this particular hydroboration reaction. That is, we say that standard Suzuki cross coupling reaction. But by using artificial intelligence, we can predict especially this particular software, OSCO software, will suggest go for alternative reaction mechanism involving palladium catalyzed carbon insertion. So here, instead of procuring this chemical, this building block, this may be expensive. But by using our software, ASCOs will suggest use of these two reagents. So they may be cheap, and the reaction will be faster and give better yield of this product. So which pathway we need to go, we can get idea from the software which works based on the deep neural network. So it will give suggestion that we can go for palladium catalyzed carbon activation. It can use high temperature. Otherwise, you can use polar aprotic solvent like DMF. Okay. So this type of condition can be prescribed by the software. We can use it, and the researcher obtained a yield ranging from 39% to 97% of yield by using this reactions suggested by the software, instead of going traditional Suzuki cross coupling reaction. This is traditional method which requires vigorous reaction conditions, and it takes 1 day processing. But this carbon-carbon activation will complete in few minutes. Okay. So third step, it is forward reaction prediction. To ensure algorithmic synthesis design robust and actionable, and it must be really applicable. We say that the synthetic reaction decided by the software, it must be feasible for us to do it in the lab. So this can be further validated. We need to study the literature and get thorough knowledge. Then we can apply the reaction route suggested by the software. So to speeding up new drug discovery, so this is the simple diagrammatic presentation. Same thing, what we take the possible protein targets and we remove incompatible drug structures or toxic structure, and identify only suitable structure based on the neural network programmings, and analyze it. And if we find a good drug, then it will go for further development. Otherwise, the cycle will continue. Okay, what are the challenges in drug discovery while using artificial intelligence? Companies who use AI technology has to go through vigorous process to copyright their work so as to secure patent rights. So to patent a drug structure which is suggested, which is identified through this artificial intelligence based prescriptions, it needs to go vigorous, very lengthy process for the copyright. We need to develop a program, then apply the program to the drug design and synthesize. Then it can be evaluated pharmacologically, clinically, it become a drug. To patent this drug, we need to take copyright of all the programs developed or modified during the drug discovery projects. So vigorous process is required. Security is another problem. We know that electronic gadgets can be hacked or can be corrupted. So security, data maintenance, data security is the major concern. So it is, say for example, if we take a patient data, so we need to take genetic level information. So when we take a genetic code from the patient sample proteins, so legal issues are involved. So that is another problem, another challenge. And faster computation is required for handling big data. So suppose we want to handle such a huge data, we need to make a computation rapid, very fast. So currently whatever computer they are using, supercomputers, it should be replaced by quantum computers. And still no success story where a compound generated through AI technology. But I am going to highlight the company In Silico Medicine. It is a biotech company. It proposed new target protein and new inhibitor of that target protein. Okay, so this is this company discovered or proposed target protein, and the drug bind that particular to that particular target protein. Okay, for which disease? Idiopathic pulmonary fibrosis. So both were identified through artificial intelligence based tools. Okay, and they have applied investigational new drug application last December. So 6 months ago, the company In Silico Medicine applied for IND, investigational new drug application, for as they claim that the identified compound is effective uh, and safe. In the early, after couple of years, they are going to start clinical trials. So these are the challenges in drug discovery. And the future problems, as we say that new and re-emerging infectious diseases like COVID-19 or mutant strains of COVID-19, secondary fungal infections like white fungus, black fungus, they are coming, and currently available drugs will give side effects. And healthcare budget is rising for almost all the countries due to pandemic, and a growing number of elder people, and also lifestyle related health issues are there. So these are the future problems we are going to face. And what are the possible solutions? We need to find new drugs, or we need to find the vaccination program. We need to facilitate the vaccination program, and to tackle the strains, newer strains, identify new drug, new medication with low side effect. And we need to integrate current practice with current science and technology. Integrated research is very much needed at this point. So pharmaceutical chemist, pharmacist, computer engineering, and artificial intelligence scientists, so all must integrate together, come together to find solutions for these the future problems. So I would finally say that artificial intelligence is a key to the future. So why we say that? When, okay, so I will finish with the story. When we talk about, when I saw a YouTube, there is a interaction or interview between Elon Musk and Jack Ma. Elon Musk, he is a founder of Tesla companies. Jack Ma, you know, the Amazon CEO. So Elon Musk strongly believes that future will be artificial intelligence based tools. Uh, so he predict that coming 20 years, there will be a transformation. All the science and technology will be moving towards artificial intelligence, and it can save the time and other economical aspects. So final word, I would say that artificial intelligence is a key to the future. Okay, these are references. I acknowledge our collaborators from UCL School of Pharmacy London, SMCRG, CHPC, UKZN, University of Free State, South Africa, and the National Research Foundation, South Africa, and the Durban University of Technology, our collaborator in South Africa. Special thanks to the chairperson, the director, the principal, Roland Institute of Pharmaceutical Sciences, research coordinator, Professor Visumohan Sahu, organizing team members of this online faculty development program, AICTE-ATAL Academy for sponsoring this faculty development program, which is highly precious for all the faculty members. Thank you. Thank you all for listening to my presentation.
Thank you, sir, for your nice presentation. So we have received some questions in chat box. So we can check now, sir.
Another question, can we do the ligand-based study with Python instead of CAD? So Python is a program in the CAD say that computer-aided design, it is a broad terminology. So Python-based programs are used in both the method, ligand-based method as well as structure-based method. So we can do the ligand-based method also. Yes. We can do both the methods. Another question, what about the side effect studies? Yes, we can predict the toxicity prediction. We say that TOPCAST, TOPCAST, that is the program which can predict the toxicity. How they predict the toxicity based on the values. If you say if the MDMK studies or PK studies, there is a ideal value range is given. If the predicted value out of the range, we can say that it could give toxicity. So we can find which group is responsible for the toxicity, and we can modify the group or the functional group in the chemical structure. We can predict. Yes, software ASCOS is open source. It is available initially free for academics. You can take it, make a trial, and say 1 month trial is available. You can use it for trial basis. It is available. You can do the prediction of synthetic route as well.
Thank you, sir, for collecting the artificial intelligence in drug design and drug synthesis. So I think it will be very useful for the faculty members and the research scholars who are involved in these research activities. Thank you, sir.
Thank you. Thank you.
Thank you so much, Dr. Chandrasekharan sir, for sharing your valuable experience upon artificial intelligence and drug synthesis. This is for sure going to help all the research scholars to upgrade their research scheme in artificial intelligence module. May I now request our principal and convener of this ATAL FDP program, Professor Dr. B.V.B. Ravikumar sir, to felicitate Dr. Balakumar Chandrasekharan sir with an e-certificate of appreciation as a token of our gratitude and respect.
Thank you, Dr. Balakumar, for accepting our invitation. And I think this is the third time you have enlightened all our participants with valuable information. And this time, the advancement of artificial intelligence in pharmaceutical field. I'm very thankful to you. And on behalf of Roland Institute of Pharmaceutical Sciences, I would like to present this certificate of appreciation. Please accept it, sir.
Thank you, sir.
Thank you so much, sir. Thank you, sir.
Thank you, sir. It's a pleasure to be with Roland for the presentation. Thank you.
Most welcome, sir. Dear participants, with this, the session 3 and day 1 of ATAL FDP program has come to an end. We will be resuming the day 2 of the program tomorrow sharp at 10 a.m. on the same platform. Thank you all dear participants for your kind attention and your active participation. The feedback link and the attendance link for this program will be provided right now, and it will be activated and it will remain active for the next 1 hour. Please fill the attendance sheet and your feedback, and please provide your valuable feedback also. Thank you all the participants for their attention. Thank you very much.